And so we're going to get started first with Gil Nelson, who's the director of iDigBio, and then Dave Blackburn from the Florida Museum will follow Gil. Thanks so much, everyone, for being here.
Thank you very much, Jill. So I am excited beyond belief about this workshop and the opportunities that I think we have in front of us. I remember 14 years ago, 13 years ago, whenever this whole APC digitization effort was launched, we were very proud to be taking 2D images and seeing people take 2D images. And we were very proud to see those images get better and better. But I really think where we have to go in digitization of collections is in other types of media and other types of imaging and recording of information. I remember going to visit Mike Webster up at Cornell, and they started their project and didn't know if it was going to get funded because it seemed to be out of scope, out of line with what folks were doing, but they did get funded. I remember being at a meeting with Dave Blackburn where they basically took over the workshop at the end, and we came out with a proposal for OBERT. So I think all of these imaging modalities are fantastic for us. I think it helps us integrate our data. I think it helps us help the community understand what biodiversity collections are all about, what museum collections are all about. And I think this workshop is really sort of the launching point, as I see it, for bringing a lot of this together and making these types of images and types of media that are available out of collections more current and more available to everybody. And maybe most important, how do we encourage others outside of this room who are not experimenting with photogrammetry and all sorts of 3D and CT types imaging? How do we encourage folks outside of this room to take that up for their collections and not see it? I remember when we were in the workshop that Dave took over. Dave, I hate to say it that way, I didn't mean you were rude, but you did sort of take it over. I mean, the second half of the workshop became David. But the bottom line is, you know, how do we, I remember sitting in that room and folks saying, can we do this? This is very expensive. How do we do CT? I mean, CT costs a lot of money, So we began to look around the room, and there were about 25, 30 of us there. And we identified 8 CT machines out of the 25 or 30 people that were in that room. And then we began to realize maybe it wasn't so out of bounds to do a CT type of workshop and to really begin moving that direction. So very proud that that happened. And I think we've got, this is a catapult for us. So I'm very, very glad to be here, very happy, delighted, and want to really hear what you guys have to say. And let's see how we can move this forward in the community. So thank you very much.
I'm just talking. That's it. They're just going to be done.
Yes.
Okay, cool. Just making sure. I don't normally do this, but this is how I wrote my notes. All right, where is the camera here?
I know it's a little awkward.
That's fine. We'll just start. All right, cool. It's nice to see so many of you, John. It's been like years. So good morning. I'm happy to begin with a few comments. That my intention really is to inspire you as you think about today and as you get together in breakout groups tomorrow. So today you're going to see a lot of fantastic images, but not in my talk. I don't have any slides, and that is completely intentional. So before diving into methods, I wanted you to take a step back with me and think about why we're doing what we're doing. So you're going to hear from leading experts about advances in creating new media to see the outside and the inside of animals, both alive and dead, both from the past and the present, as well as, thinking about imaging the places those organisms are from as well. You also hear about these cutting edge techniques like some that Arthur is leading to make those images and data useful in research and education outreach. But here's what I want you to remember most. When we do this work as part of the Natural History Museum community, we don't do this to create images and data for their own sake, right? We do this because we're intending to serve some purpose, or more specifically, someone. And though today will be about creating images, I hope that you don't lose sight of the fact that there are people who we intend to see and to use these images. And those are the people that we serve through our work. So many of you know that the Open Vertebrate or OVERT project recently concluded just a few weeks ago. We published our sort of culminating paper in Bioscience detailing the project. And so if you're interested in that project and don't already somehow know about it, please check it out. That is the culmination of work across 25 institutions across 6 and more years, involving more than 250 different individuals that helped with some component of that project. So it's really a community effort. If you read it, you will see that the first paragraph says nothing about CT scanning or imaging or digitization. The first paragraph begins with this bold claim. The physical specimens and objects in natural history museums require people to give them meaning. So what I've learned most from leading OVERT in collaboration with many of you that are already here today is that the most interesting part of the work really hasn't been the images, it has been the people. And there are people searching for our data. There are people who are sending these things on loan, generating data, making images, sharing them online, using them in their work. There are people that are using them in research, but using them also in classrooms for home 3D printing projects and more. It's also the people who have given us feedback about how valuable all of this has been. At the very beginning, in this workshop, I guess I took over, there was skepticism that anybody would even want this sophisticated of data. Like, is this just overkill? People really could just use 2D images. Do we really need all this more data? And it's really from the people using these data that have shown that, in fact, yes, there's a market for this. There's real value that people ascribe to having access to these type of data. So to me, one of the most remarkable parts of OVERT, and by extension also Morphosource and others, is that we're now better able to see the who associated with parts of our data. Which institutions and individuals generated these data and shared these data, manage these data, download and use these data. There's actually people and even names associated with all those pieces, right? And I think if you look back to a flat data record 10 years ago, there's not really that many individuals in that data record, right? And now we actually can keep track of that. So the numbers from the OVERT project are fun to give, so I'll just give them now. So we scanned more than 13,000 specimens together that represent about 6,500 genera. That's about 2/3 of all living vertebrate diversity. Genus diversity. Those are represented now by about 30,000 media files on Morphosource, and that have been downloaded at least 100,000 times. They've also been viewed well more than 1 million times. We have a hard time keeping track of like views and who those people are, but we know that they're getting viewed a lot. And you can, quote, use those data just by viewing them. So with a new viewer that was embedded into the Morphosource website, there's a lot of science that can be done straight off looking at the data online, right? So those views are important. The goal of NETCN, like OVERT, was to generate data that was for research. And so no surprise, that's happened. So there's been more than 200 publications that have used OVERT data. I stopped counting like almost a year ago, so hopefully it's much more than that. And those have had nearly 700 individual authors on those papers, right? So there's a lot of people out there using these data. It's difficult to know how many people are viewing the data online, but there's at least 4,000 different individuals that have downloaded those data for their research, their classrooms, their art projects, or their home 3D printing projects, right? So it's clearly reached a lot of people. So as you're thinking today about imaging, I want you to think too about the people connected to it. You know, it's easy to imagine a generic researcher who will want to use these data and images in their future. And a lot of digitization is kind of like that, right? We're doing it, we're imagining someone's going to want it in the future. But I find it helpful to actually think about specific individuals and who that person might be and what they might want. And while we're not yet at a point where we can probably have these detailed 3D data for everything, and we probably never will, you know, by not imaging everything, it means we have to make choices, right? We have to decide what we're going to image. And I think thinking about the people we want to reach is helpful for thinking about the choices we make while we're doing imaging and digitization. So, you know, how will a 5th grader find and use these data? What might she want to discover? You know, how will a high school environmental science teacher find and use these data with their students? And what do they need to know in order to do that, right? What type of things are going to be interesting to a digital artist in Australia or a sculptor in Korea? Those are people who actually use our OVERT data right now. Ed and I always talk about what's going to be the 1 million download specimen. You know, can we predict what that's gonna be? Can we make that happen? That really depends on what people want. So we can't be all things to all people, but I find it really useful to imagine at least a few people that help guide what we choose to image, how we choose to image it, how we choose to share it. You know, I've been asked many times what Overt Part 2 is. To me, that is the Nocturne project that Ed is going to talk about later today. So we already know pretty well as a group how to make data, how to make images. But the next piece is actually, I was really happy to hear you say that, Jill. The next piece really is the community of people, you know, that we want to bring into the fold and how we have them work with us. So, you know, it's enticing to think that a greater diversity of people will find and use our specimens because they're online. But I don't think it's sufficient to just put things online and hope. I think we actually have to put more skin in the game as a community to identify, you know, who those people are, what are their obstacles, what do they need to know that we can help them to make use of these data. I think we have to actually identify specific groups of people, regardless of what that means, in order to do this work better. So, you know, I have no doubt that technology is going to change, and in 20 years, this would be a very different workshop. But I actually expect that the people part of it will be pretty much the same. Right. And so I think, you know, taking the opportunity now to think about those people and what they might want and what they might need is valuable for us as we begin making decisions about the specific things that we're going to do in much more detail with 3D imaging. Right. We can't do it all, but we have to make some choices. So who are the people we want to serve? And I'll just close to say that, you know, I hope the words "who for" sticks in your mind today and tomorrow. You know, I think as part of being a natural history museum community, the goal of what we're doing is to do this for other people and for other institutions around the world. And that could be scientists or teachers or fifth graders. And that is the lesson that Overt taught me, which I thought was really a project about CT scanning dead fish. But ultimately really showed me that it was a project about, you know, connecting to a bigger diversity of people that wanted to make use of the things in museums. And so that's what I wanted to share this morning. And I hope that sticks with you today. So, thank you.
Thank you.
All right, so next we have Nelson Rios from the Yale Peabody Museum. Give me a second to pull up Nelson's presentation.
Is it in PowerPoint?
Pointer.
There you go.
All right, thank you. So I'm going to talk about a instrumentation that we built for doing 3D or multi-view imaging. I'd also like to just point out Victoria Tran, who is my co-author on this talk. She produced a lot of the material and images and does a lot of our 3D scanning at the Peabody. All right, so just to give a little background, this project dates back many, many years, going back to 2010. When I was at Tulane University working with Hank Bart, we had a project where we were trying to do 3D scanning of fishes using a laser scanner that was fraught with all kinds of problems, lots of limitations in scanning fluid preserved fishes with lasers. And so we started to look at alternative techniques. We came up with photogrammetry. But one of the problems was scaling it and being able to do it in a highly repetitive nature. And so that led us to develop this prototype instrument that you can see on the bottom left of your screen. And it's basically just a bunch of cameras that move around and take a bunch of pictures that we can then use for doing 3D scanning. But that was just proof of concept type stuff. We never actually went to the next stage of getting a real full-blown project to do large scale scanning. So fast forward to 2019, at the Peabody, we got a project from the National Endowment of Humanities to further improve upon this infrastructure and actually do a production level scanning project. And so we scanned a number of materials from the Yale Peabody Babylonian collection and the anthropology collection. And then finally, now what we're working on now is using the same instrumentation to be able to do the same kind of concept, grabbing multiple views of images, but we're working with one of the more challenging critters to be doing this with, insects. And so we're using this very big device to image lots of little tiny insects. We're trying to see if we can reconstruct 3D data, or at least identify what this is applicable for doing 3D scanning. But also we're doing it to do these really rapid data sets where we just extract the label data from these multi-view image sets. And there we might grab only a dozen or so images per specimen. So the project's been going on for many, many years. I've got long slew of collaborators that have contributed. One thing I like to point out are all the names in yellow and in blue. Those are the number of students that have worked on this project. A lot of this work has been done by students. In fact, some of the software development has even been led by students early on in the project. So here's just a sped up view of what the current instrumentation looks like. There's normally two chambers that would be stacked on top of each other, so you could do full 360 in one go. But this is the setup that we're currently using at the Peabody, where we're using it on a big table, and it can go around. And you can see on the other side there, the specimen rotating around that was actually scanned as a result from this. Let that finish playing so you can see what it's doing. The cameras do this little 360 spin. This is a very old version. The current hardware actually doesn't do that anymore. It's much more optimized and can now do a lot of rotations without having to worry about wires tangling and things. But those were little things it was doing to prevent wiring issues. On to the next slide. So here are some actual scans of some biological material. A number of insects we've scanned with the lightning bug project, small fossilized gastropod that was on loan to one of my colleagues that's working on that specimen right now. And you know, with the insects, you can see there's pins in them. All of our scans, we usually do them with the pins and the labels. Our goal is not necessarily to just do 3D. We want to be able to do 3D at scale. And so the idea of taking off pins and all that's too much work. And so if we have to program the cameras to take more photos so that they can get the right angles and get in there and reconstruct it, that's what we try to develop. We've played around a bit with how best to do pinned insects. We've come up with this kind of little jig that you can put a specimen in and have the different markers embedded on it, still pretty small, and allows us to get to be able to reconstruct specimens pretty well. We have a pipeline that's mostly automated, so we've built this software for operating the machine, deciding where we want to position the cameras. The idea is a technician can design a particular path that would work for a range of specimens, and then she just has to put a specimen in there, press a button, and the imaging happens. But we don't want to have to then download this gigantic data set off the cameras. And so the idea is you just spend all day imaging. And then overnight, we do this bulk image transfer off the cameras. If there's 3 cameras, 6 cameras, it doesn't matter. All the images come off. And then once the images come off, we run it through a pipeline of various scripts that do various things. We actually associate the pose metadata, the positions of where all the cameras were, with all the images. We create these overview sheets so that we can see what the imaging was done, and we can get a very quick assessment of how well the imaging was done. Then we actually configure these manifest files that we've created so that we can automatically pipeline all the photogrammetry. So we actually don't do photogrammetry per se, where you're sitting there and doing it manually in any software. We just run a script and let it go through, and then the next day we can look at those results and say, hey, did this 3D come out well? If it didn't, maybe we want to tweak some options or whatnot. And then we get the output results. We also take some of these data for the lightning bug project, and then we feed it to our colleagues at Argonne National Labs, where they're developing the algorithms to extract out label data and produce these virtual labels, and then eventually structured data out of it. So this is the kind of pose metadata that we get out of it, you know, for every session and camera, we have the coordinates that come out of it. But being able to associate this information with these cameras, like I said, we're just doing this imaging where we're plopping, you know, things and everything's getting stacked up on a big SD card. And so we have to have a lot of trickery for being able to then, after it's all downloaded and be able to merge it all back together. And so we rely on a lot of the EXIF data and being able to do the timestamps at which the images were taken. The software records every single time an image is taken and how long it takes for that image to be saved. And so we know a start time and an end time for every image. One thing we've learned, if anyone wants to do a similar thing, is that camera clocks are very inaccurate. They tend to lose time. And so we do have to calibrate the cameras every couple of days to make sure that they are in sync with our computers. Otherwise, they will be off by a couple of seconds. And a couple of seconds when you're doing high rapid imaging makes a big deal. These are the composites that we'll generate, so we can quickly overview. We put the coordinates and which camera did the imaging. And that just gives us a quick view as to what was done. This is a manifest that we would then feed. We do most of our 3D reconstructions using a software called Reality Capture. I think there'll probably be talks later today that focus on that piece of software. So we can specify all the various settings that we want, the targets we want to hit. And this is a manual creation. We haven't automated this part yet. And so the technician will sit there, edit this file, dump it into a folder. Once it's in that folder, another piece of software is just sitting there monitoring this folder. It identifies there's a new file in there, and then it just feeds it right into the photogrammetry pipeline. We also have the ability to do focus stacking, which I'm seeing here, right here, this little line, where we can run it through Helicon Focus. If we use Irene Stacker, which sometimes we do, we'll do that manually. Oops. Here's an example of focus stacking. We don't really try to do focus stacking because it changes our scalability significantly. A focus stack data set could take us 5 hours to produce. So we try not to do that. This is just a test of the kind of quality we could get. So if we're doing like a Z stack, this would be very similar to how like a stack shot works, right? It's a camera moving along a lead screw. So we can get really good quality, but we also wanted to be able to test that we could run the Z stack using all 3 axes of the machines, which have different mechanics involved in making sure we could still get good data out of it. So here's kind of how we would do label extraction. We would have a data set, and obviously there aren't enough poses in that data set for doing 3D reconstruction, although maybe you could have on a very coarse level, but the idea is just to have enough images so that we can see from various fragmentary viewpoints all the different the information on the particular label, and that's used for reconstructing. Right now, we're actually overshooting, taking much more data than we need so that we can work on developing the algorithms and have what we need. So we can also do things where we take a number of specimens and just line them all up and then build a path that's kind of designed to encompass all of that. And then we can do 3D reconstruction of multiple sections at one time and then just segment it out. Whole drawer imaging, this isn't something we've really done a whole lot of. It's something we want to explore a little bit further, seeing what kinds of data we can get out of it. Here's a drawer that we did scan. It was mainly just as a proof of concept saying, hey, we can do it. But it's something I think we still need a lot of work. The drawers, the size of the cartons and all that really become problematic. And then if you want to do more traditional type turntable type scanning, we have little widgets in the software so that you can take the same software and take the same hardware and kind of just plug it up a little bit differently and use a couple of different components so that you could operate a turntable, or you could operate the camera in kind of like a 360-degree panning mode if you wanted to do something like that. The hardware, like I said, here is like a 2-chamber setup, which is what we have set up at the MCZ at Harvard right now. This is what we have set up here at the Peabody, and these are with the electronics and the little camera cradle like. And it's all open source hardware, so if anybody wanted to replicate this or contribute to it, you could just go to GitHub, download all the designs, and start rocking and rolling. The software is also on GitHub. You can see here's one of the more complicated paths we've created. It uses 3 cameras going around doing this kind of elongated ellipse around an object. And like I said, there's lots of different tools for automatically creating these paths. You know, this one is this one that you see on the bottom left corner is probably the most efficient one that we've been using lately because it makes it easier. We can just take the approximate measurements of the specimen, the length, the width, the height, punch that into a little calculator, and then come up with this little path that will make sure that the camera is always a certain distance from where our plane of focus is. And that allows us to not have to rely on autofocus and things like that. And for insects, autofocus tends to become very problematic. So our next steps, as far as what we're working on, or these aren't necessarily things that we're specifically working on, but things that we would like to tackle at some point in the near future, or if there's someone who would like to collaborate and take on some of these tasks, we'd certainly be open up to it. One would be adaptive lighting. Lighting is still one of the challenges we have. We have all these cameras moving around and you want to avoid hot spots. And so having ways to have the lights be as smart as all the other elements of the device and say, hey, well, this light should dim out or turn off or do whatever. And so that kind of stuff we want to explore at some point. Preliminary object detection using techniques like VSLAM or something to say, oh, there's an object in the thing, here are its approximate dimensions. We'll just automatically create a path around that and then do the high resolution imaging. Integration of structured light, that's something we've been wanting to do for a long time. It's been on the back burner and hasn't had a real priority. Using the data from the pose metadata to create camera priors that we can use to assist photogrammetry, doing alignment. Other things where we do the masking in AI as opposed to relying on our current process for masking is to actually reconstruct a 3D model and then spit out masks from that 3D model and then redo the 3D model. But there might be other alternatives. Some of the early tests we've done with using like Segment Anything have been phenomenal. And so I think this is really a good opportunity to tackle some of the masking things. And then ultimately, increasing our documentation and community engagement. It's very easy for me to say this is open source, it's on GitHub, but without that documentation, it's not going to get a whole lot of use outside of that. And so that's something we're really interested in doing, is improving the ability to use this and have other people either develop it or use parts of it to do whatever they need. And with that, I will end. I hope I finished on time.
We're ahead of schedule.
Am I?
Yeah.
Oh, thank you. I was a little worried I had too many slides.
Perfect. Thank you. Thank you. Nicole, you should be able to
You'll have to
share. Oh, okay. Maybe she could share over it, but. Okay, and our next presenter will be remote presenter Nicole James from FSU. And again,
Hello, thank you.
yeah, again, just a reminder, if you can put questions at Q&A, you can put that in at any time, and we'll address those, I guess, after our next speaker. Perfect. Looks good.
All right.
Take it away, Nicole.
Hi, thank you so much. My name is Nicole James. I'm a digital media researcher with iDigBio at Florida State University, and I have been exploring and utilizing 360-degree cameras in the field for capturing specimens of varying sizes. So I want to talk a little bit about 360-degree cameras as an omnidirectional multi-tool. So I want to contextualize 360 degrees first as a type of immersive media, and that simply means that this is media that leverages the 360-degree space and allows users to experience and interact with virtual content. And this can include camera captured content from 360. So immersive media simply creates a more interactive experience, and this can be accessed through mobile devices, virtual reality headsets, or physical sensory spaces. And users get the impression that they are embedded within the scene, and this is accomplished through something called degrees of freedom. So your 3 degrees of freedom allows you to look around a space, while your 6 degrees of freedom allows you to look around a space, but also approach and get closer to objects in that space and interact with them. And 360 is often compared with virtual reality, but this distinction between degrees of freedom is where they start to set apart. So the 360-degree experience is limited to the movement and location of a camera. So there's only 3 degrees of freedom and limited viewer interactivity with a scene or space. And this includes the length of a video. So rather than a computer-generated 6 degrees of freedom environment, 360 immerses the viewer in a real-world video or photograph. And this camera captured content can be accessed through virtual reality headsets, but it's not required. So you can access this through your mobile device, that physical space, or simply through online host sites that have navigation tools embedded within them, such as YouTube. So here's an example from the Florida Coral Reef diving experience. And you'll notice here that I'm able to click and drag the frame from left to right. And we can notice that we're moving with a marker. So we are moving with the camera in this coral reef, which is operated by our diver here. And while I cannot access or get closer to the schools of fish or go out and explore the reef, I can at least reorient my camera to what I want to focus on. So what is 360 adding here in addition to our traditional imaging? Well, if we were to capture a habitat such as the reef, we would have to take an image in a single lens mode like so, then change the angle, take another, change the angle, take another, and then eventually make it all the way around and then compile a sense of that space. And while this can bring helpful documentation, it's inadequate for capturing the kinetic nature of something such as a coral reef or an active environment. So the omnidirectional shooting of 360 grants a more accurate representation of the biodiversity at a single moment in time. And many 360 cameras are able to vary their capture mode. And because they were originally designed as action cameras, you can see here, this is fully waterproof, and most are, but at the very least, they're water resistant, they're durable, which makes them well designed to be taken out into various terrain for environmental capture. If you did have a headset, which is nice, you could look around this space from YouTube, but because of that variable mode of being able to view 360, you have different options depending on what technology you have as a user. And what's really great about these cameras for capturing this content is that they are largely cost-effective. So currently I'm using a consumer grade camera, the Insta360 X3. This is a completely waterproof camera that can be submerged 33 feet in the water. It can shoot in 5.7K for 360. It has built-in stabilization and stitching. And many of the consumer grade models will have comparable features as well. Once you start getting into professional grade cameras, you can range well beyond $5,000, but this would include your 8K shooting capabilities and above, larger camera sensors, deep dive capabilities, and more than 2 lenses on the camera. So 360-degree cameras capture their content by combining imagery and video from 2 or more lenses. And I mentioned internal stitching. So this is the 360 that I've been using, this X3. So you'll notice there's a lens on either side, and then it would stitch up the middle. And so this camera is really compact, and I was able to take a simple camera clip here and attach it to the inside of a cherry blossom tree in bloom here in Virginia. And I'm able to take a look at the active pollinators flying around from blossom to blossom. And what's really nice about these 360 cameras is that not only can they shoot in 360 like so, but they are able to shoot in different modes, such as a single lens mode, time lapse, and they include similar setting changes to traditional cameras, like changing your frame rate, your resolution, ISO, white balance. And they also often include specialized modes for tracking GPS data, or you can connect them to your mobile device to control them from afar, which is what I'm doing here. So I was able to place the camera in a really interesting point of view from the inside of this tree, step away, and allow the pollinators to come back and fly right by the camera. And this here is an example of a still extracted from this video. So this is what 360 looks like stretched out. So you can see where this gate would connect over here to the other gate to create that single frame. And these cameras have been used for a number of research applications, including reconstructing 3D models, mapping route behavior from the back of a shark, conducting non-destructive monitoring of species, and applying this data to conduct observational training in virtual reality headsets. And these images have also been combined with machine learning to help detect plant growth, detect trees at field sites, and entire classes and events have been documented to be experienced in 360, which makes them more accessible and able to be revisited. And this data has also been used in architectural conservation to identify and monitor deterioration mechanisms. And this can include using 360 images as a base for a virtual tour, where you can embed information such as 3D models so that you can use a more active site within 360 to share embedded data. And here's an example of this from the University of South Florida Center for Digital Heritage and Geospatial Information. So I'm able to look around the images and anchor points within this virtual tour, but I'm also able to click on these animations and interact with embedded media. So while using these as a host, I'm still getting an idea of the space, and I can also look at more spaces within the map here in the top left corner. But this can serve as a host where you can interact with 3D models, text, audio, standard video, and images, all while situating this information spatially within a given environment. So even though we're remaining in our 3 degrees of freedom, we are adding interactivity for not only outreach, but this could also be used as a virtual field log that could help contain and visualize research information. And what I've been working on is I have been applying 360-degree camera use for capturing specimen collection sites and experimenting with best practices for utilizing these devices. So I've been mostly focusing on capture methods and patterns on the ground to best document varying sizes of specimens, such as small flowers to these 2 trees in bloom. So I've been exploring how these video and images can not only add missing context, like situating them inside a tree within the cherry blossoms, but also using this visual data post-capture. So this would include the 2 3D models you see here that I've created via Agisoft Metashape. So I've taken stills extracted from 2 360 videos, and this is one of them, to then create a virtual model. So I'm leveraging our 3 degrees of freedom here in our 360, and then I've even converted it into 6 degrees of freedom with our 3D models. Rough 3D models, but still gathering that spatial data. And while 360 is great, some challenges in utilizing the cameras include that imperfect stitching. So the more lenses you have in cameras, the more problems. I've noticed this in my work, particularly with complex patterns, overlaps, or when they are closer to the camera, it starts to struggle with that stitching here. As I tried to capture this cardinal, you'll notice those branches didn't quite overlap perfectly, so I'm working through best practices to avoid that as well. And common issues with 360 overall include file size and hosting needs for the files themselves. This technology is constantly updating, improving, and changing for better or for worse, but that also means needing to update and acquire proper software to view and host them. And some consumer options, including the X3 that I've been working with, still struggle in lower light conditions, and this is partially due to the trade-off for them being more user-friendly. So some of those manual capabilities available with DSLRs are not available, or they're unable to be changed as they would be otherwise. And that is a very quick rundown and update of how 360-degree cameras can be applied to the collections community and what I have been working on here. Thanks so much.
Great, thank you, Nicole. That was great. Up next is Duncan Ershik from UMass Amherst. And again, as a reminder, you can add a question at any time in the Q&A, and all of the speakers here in this session will be available to answer your questions afterwards. So Duncan, can you share your screen?
Yep. Okay, can everyone see, everyone, I assume everyone can see that, hear me okay?
Yep, look good, sound good. Thank you.
Thank you. Great. Thanks so much for having me. This has been exciting. I want to talk today about some work on novel 3D tools for reconstructing living animals. That's been a focus of my work for the last several years. As this whole meeting is going to really outline, 3D scanning is a form of, if you want to think about it, digital heritage, is a fascinating enterprise and has many facets. There's obviously aspects of digital 3D photogrammetry, which we'll be talking a lot about today, but there's also other areas, right? There's laser scanning and white light scanning, there's CT and MRI scanning. For those of you, obviously, who've ever had the unfortunate circumstance of being told they have to have a CT or MRI scan, you're familiar with the concept that you go to a hospital, they take a scan, they create a 3D model. And so there's a long tradition of creating scans of dead objects, such as the skull shown on the left, or a brain, or a historical artifact such as shown on the right. But there's a lot less tradition given to the concept of reconstructing what a living animal might look like so that we can study it for the sake of science. Let's see here. Why is it? There we go. And, I don't really have to sell this to your group, but I think it's important to remember some of the advantages of 3D scans when we think about a living animal, which is they provide a 3D map, shape map of an object or organism. So they really do provide that ability to understand the shape of an animal in a new way. And this is going to become incredibly important when we think about certain species that really aren't found in very good systems in natural history collections. I'm not going to be talking about today, but I'm going to be talking later on in other talks about how we have some new work also that using 3D photogrammetry scans is providing a 3D map of the color of an organism. And we can map that onto the shape. These can be modified to add movement and to study locomotion. Another focus in my lab I won't be talking about today, but is exciting for the future. And the final result can be strikingly realistic. And this is very important when we think about megafauna or animals that we examine in terms of demographics, because demographics we're interested in studying particular animals and following them over time. So what about living animals? As an example of this is Mac. This is a female leatherback sea turtle that we studied in Juneau, Florida. We reconstructed this animal using 3D photogrammetry, so the body shape and color is accurate. What I admit is a bit of a flavor on our part was simply the movement that we added to Mac. But the shape was correct, and that's because we had the fortune of being able to work alongside the researchers who studied this animal and have access to this animal as well. And so what could you do if you had these kind of shapes of 3D animals and imagine a scientist we had access to these for different kinds of species? Here's a list of some of the things that our lab has thought of, but I'm sure you have your own ideas. I'd love to hear them. You could certainly collect 3D models of organisms in the field. You could get their DNA. You could release them. This is becoming more and more commonplace in regions of the world that we work in Africa and Southeast Asia, where a lot of countries don't really want you euthanizing or killing or depositing specimens or exporting them. You could perform computational fluid dynamics on 3D models of flying and swimming animals. There's a long tradition of this, but this typically has been done on kind of rough caricatures of species and not accurate models. You could use the 3D models to estimate body condition, animal health. This is one of the biggest areas of growth for a lot of our models. You could design new kinds of robots, and you could use them obviously in educational portfolios. So the 3D photogrammetry is something you'll be very familiar with. This it's already been talked about, but the idea of taking a series of photos of an object, in this case, this is actually a golden poison frog at the Zoo Atlanta with tadpoles on his back. You take a series of 2D images, you convert to a 3D mesh, and then create a 3D texture geometry. And we've been focused on creating rigs and systems that can work with living animals in the field or in the lab. Our initial focus was on small reptiles and amphibians, but we've expanded to many other animal species. You can see some of these concepts down below with some of the students I've worked with, like Dylan Briggs, Casey Smart, and Zach Corbeau. But I'm going to be talking about two larger animals that are really not that we've been interested in. One is megafauna, specifically in Africa, because we've been interested in recreating these animals to understand health and body condition. This is a proof of concept that we did to recreate a 3D southern white rhino to develop novel body condition metrics. This is Bakari. Bakari is a southern white rhino in the Perth Zoo, a very mellow rhino. We were able to actually hook up a series of cameras around this cage while it fed, and then get a really good 3D photogrammetry profile of the animal. From the 3D mesh, we could create a final 3D render with color that was an accurate metric of its shape. With the vet staff at the Perth Zoo, we were able to then measure a whole series of metrics, or they were able to do it, I should say. This is a very well-behaved rhino. I don't recommend any of you ever try this with a rhino in the wild. Body height and body length. And so we had a series of morphometric measurements that we could use to scale the model. We knew the mass of the animal at the time. And this allowed us to ultimately create an accurate 3D model of this rhino. Again, the motion is something that we added, but the body shape, the texture, everything is accurate. And by the way, I should say that we have a lot of knowledge and skill about recreating models, and we work with lots of different partners around the world on this. So if you have a project you're interested in, please come and talk to us. So the concept here is to estimate body condition from volume mass relationships. So we hope to scan other rhinos or to use this 3D model to create other digital copies. From this, we know the mass and the length of the animal. We can calculate the volume and the surface area, which is one of the exciting parts of this work. And from that, we can ultimately create new metrics for body condition that we hope to then use machine learning systems on in the wild to estimate how much these animals weigh. The second part is underwater. We wanted to develop methods to do the same thing, but with underwater animals, and we focused on hammerhead sharks, which live in Bimini and will dive with. You can dive with them fairly safely. There's a person in the middle who feeds the sharks. We have a series of cameras around. You dive with the shark, you get a series of photos, much like the rhino, and create a 3D photogrammetry scan. We weren't so interested in the nurse sharks around there, but you can see the big hammerhead shark in the middle. From this, we were able to focus on one particular individual. Her name was Nemesis. She's about 10 feet long. She's a female shark. And we're able to reconstruct the accurate body mesh. And these sharks, by the way, grow very large. They grow up to over 20 feet long, although in the wild, we rarely see animals that large. So this kind of body mesh, these animals aren't easily found in natural history collections. And this is, again, the 3D model of the shark. We added in the mouth motion and other motion, but the body shape is accurate. From this, we're actually able to actually ask some really interesting questions. One of them is that when you look at different animals we've scanned sea turtles, we've scanned marine mammals and sharks, and many other species. This is a plot just for aquatic species comparing volume and surface area. And what you see is the isometric slope should be about 1.5 and the actual slope is 1.48. So despite the fact that we're comparing reptiles, marine mammals, and fish, elasmobranchs, these animals all show the same basic relationship between volume and surface area, which is really fascinating. And we're going to continue to add to this plot. We put these models online. They are open access. At last count, I believe, actually, I didn't have time to update the slide. I think we actually have close to 180,000 downloads. They get used for all kinds of applications, from education to scientific. If any of you want to create your own model of a living animal, please come contact us. We are a cost center, so we can actually work with partners all around the world to create new content. But we do allow people to download them for free for nonprofit use. And it's been really exciting to work with lots of different people on that front. And I'll stop there. Thank you so much.
Great, thank you so much, Duncan. We have 15 minutes now for question and answer. If you want to raise your hand or add it in the Q&A. Going to have you stop sharing, Duncan.
Mm-hmm.
And then attendees, feel free to raise your hand, add a question in the chat, and we'll be watching both. We do, yes, thank you. Okay, we do have a question here in the room from John Woodward. Go ahead, John.
Question for Duncan. How was the motion added to the 3D models?
Right, so we actually have developed a machine learning algorithm that can take motion from an animal and put it into a model. We published that 2 years ago, and I'm happy to send that paper to anyone who's interested. But for these particular models, that was honestly just for fun. We just added the motion and basically our knowledge of fish, shark locomotion. So we actually create both static models and also we create just animated models, and the animated ones primarily get used for education, but the static models are the ones that get used for sort of research and applications and things like that.
I have a couple of, does anybody else in the room? Beth Brainerd.
Yeah.
Yes, but there's a couple questions.
Yeah, there's a couple of follow-up questions to that one. So Alana Rivera asks, how much of your work relies on a still or relaxed individual?
Yeah, a lot of it, yeah, absolutely. Like in the case, but not always. Like in the case of the hammerhead shark, they were never still. I think we were filming at video. I think we were filming at 120 frames a second. So in the case of you actually can use video as long as the quality is sufficient of the video to recreate motion capture, and that's been well documented in the literature. But obviously a still animal like a frog or a reptile that just remains rock solid still is ideal. It's a little technically easier for most people, but we can do both, and we have done both, and others have done both as well.
And then another question is, can this be used to create synthetic data?
Yes, so the digital model is one of the biggest areas of growth that we've seen from other partners is now that we have, for example, 3D Rhino, there's a lot of discussion of, it's very hard to scan rhinos, as you can imagine. But there's the idea that to use 2D images of rhino that are very common to actually use that machine learning algorithms to then create lots of digital copies of rhinos from our initial 3D model. And there's a huge growth of industry of 2D to 3D. So I think there's a lot of thought about using our 3D Rhino to scale and calibrate and create new synthetic data, which makes sense because obviously scanning rhinos around the world is not a very practical enterprise. So yeah, I think absolutely that's a big growth area.
Any other questions? Doug?
Other question, so also this, this is open to all the talks, these questions, so other ones have questions for the others, just ask them. But Duncan, I was interested, so you have a lot of movement of the animal that's walking around. How does that affect the alignment when you are doing your initial alignment in the reconstruction program?
Yeah, so that's a great question. So that's actually where our team, we actually have a lot of students that increasingly are coming to our lab to learn these techniques, but a lot of the ideas about photogrammetry is with commercial software like Agisoft. But actually what we've been primarily focusing on is developing using Blender to reconstruct models. So for example, you don't really need the 3D photogrammetry software to reconstruct a shape. You can actually bring in those images into Blender and actually, even if an animal's moving, can align those photos manually. It's just a more involved technical process. So yeah, the fact that I guess in short, what we've been doing is not going to be so easy for, let's say, a high school student who wanted to learn photogrammetry. They would just more scan a rock and use Agisoft or whatever. But if you have, let's say, a shark that's swimming or a fish that's swimming, you can absolutely create a 3D model. You just have to use other software. And I didn't have time to go into it, and I'm happy to send some publications out that lay out this foundation. We do, and I should say for any of you that want to learn this process, which is not well known, by the way, I have a student who's here at my lab for a week. We have a lot of students that come through my lab and we just teach them how to do this. And usually after about a week, they figure it out. So it's a skill set we're trying to share with the community.
Michael?
Yeah, I'm interested in, this actually might be for all three speakers, but particularly for Nelson and Duncan. Both of you mentioned capturing color. These different techniques, and which is something that does not preserve well for many tags in traditional specimens. So I'm wondering, what's the prospect for doing that? How well developed is that? Do you need to use calibrated cameras? Just those sorts of things.
Yeah, I could address what we do. So, I mean, in our mounts, when we're doing 3D imaging, even when we're doing, less dense image sets, we always put a little color checker in there, a standard color checker, so that we can calibrate. We do most of our shooting in RAW, and then we can do the color correction afterwards. It is an extra step in the process to do that. But yeah, that's what we do.
Yeah, I didn't have time to talk about it. My graduate student, Josh Medina, has been developing a way to quantify color analytically from photogrammetry data. It's quite an involved process that he's been pioneering with birds. And the key thing is it's fairly straightforward just to get color data from photogrammetry, but it's a whole nother thing to develop a kind of a whole analytical package around it. So that's what we've been working on, but we haven't published that data yet. And he'll have a paper he'll put into review in the next month or so that'll address that. I'm very excited about it because I think it's actually allows you to map on color data onto shape data pretty accurately. I think it'd be for collections, it's going to be fantastic. So.
And we have, oh, sorry.
No, Nicole, did you want to add anything? I feel like we're dominating the conversation here.
Oh, no, it's totally fine. I'm not using currently color checker data right now in terms of the in the field surveys of using 360. Particularly because I think it'll be more difficult as well to find color checker. I've actually found color checker data with the flattened out images to be a little bit more difficult to apply. So that is something we're also working on too.
And also for you, Nicole, have you played with underbrush or forest floor for omnidirectional work? The person who's asking the question is curious if there's any sort of environmental density challenge, for example, if there were branches in front of a cardinal.
I have not played with underbrush or forest floor for the omnidirectional work. We've been focusing largely on capturing actually a large portion of a field site too, and working to see what ground pattern that we can get perhaps the most accurate representation or something that's a little bit closer to accurate. And I think I can see there was another answer maybe or a question about accuracy as well. But yeah, we have been largely trying to see if we can capture the multiple sizing of specimens in a space. And there's been really interesting interaction between the density challenges as well. If you noticed in my slide, I did have 2 layers of the 360 camera. So I had it at about this high, and then I used an extension that reached it higher, which makes up for some of that overlap. But there's still kind of those struggles with overlap and environmental density too.
Thank you.
So there's been some interesting work done in Cork, Ireland. I cannot remember the researcher's name, she's a paleontologist, and she has tried to reconstruct color from fossils. Yes, right, and pretty fascinating and more challenging. They're trying to reconstruct from, I think, even specimens. So I don't know how she's doing that. Also, back to John's question regarding movement and how movement that was shown on the Leatherback. Beth Brainerd at Brown has done a lot of work with birds and trying to understand the internal anatomy and how birds function through adding to specimens movement from bird images.
I have a question for David. So I really like the talk and I like the board. So it's such a big, diverse audience that you're maybe not even having in mind at the time that you're creating the models, but instead just creating the models and seeing who will come. Am I wrong in that? Is it the case that you've identified from the start these 5 groups that are personified by the artist from here and the 5th grader from there, and how do you think about your audience?
Yeah, I think it's actually a really interesting question. I don't think it's something that we went into it thinking hard about, other than just thinking about scientists. Scientists want these data somehow and they'll be useful somewhere else. But yeah, I think it's partly from the fact that I've approved so many thousands of download requests that, those all come from people with names and they write about what they're going to do it. So I think that's part of what's kind of made me think hard about it. And Ed and I have talked a lot about, using the download information we get to think about like, well, what is it people really are gravitating? Like as systematists, many of us are in the museum community, we tend to think type specimens. But it's like, well, I don't think type specimens are really that useful to people outside of systematics, right? Usually like they're the worst ones. But I do think that, like I'll just give an example. One of our most used specimens from over was a WIM. It's not even a fluid preserved specimen. It was a skull of a gharial crocodile that one of my grad students decided to just put in the scanner and put online. And it gets used constantly for science and outreach around. And that just has made me think, when we talk to a lot of collection folks to pick out specimens, you know, there's a lot of specimens that would meet our criteria, like, all we need is one individual of a species of a genus, like, we have plenty of individuals, you know, of an anole. Like, which one are you gonna pick? The ones that we would often try to gauge or guide people to is ones that might have some other story of interest outside the research. You know, like the last one ever seen in place X. You know, I used to joke that if you got a choice between picking a specimen collected by Dave Blackburn or Charles Darwin, like you'll always pick Darwin, right? Because there's gonna be school teachers that are suddenly interested in it, right? Because of that. So I think there are audiences, you know, we tend to have this, maybe sometimes it's explicit, most of the time it's implicit, that we think that somehow the work that we do related to digitization kind of connects to DEI issues and accessibility. You know, more people can now use the stuff behind the scenes of the museum. But you know, our sort of brief foray into looking into that based on user data doesn't suggest that's true. It's basically just more of the same people we already interact with, which is fine. But at the same time, you know, it kind of speaks to where if we're really the goal is to reach outside, you know, that community with these data, then we need to be a little purposeful. And I think going through usage statistic data can get you some sense of like who your users are and how you might make things different. Ed or Jamie, we've talked so much about this. Do you have thoughts?
No, I mean, I'm gonna talk a little bit about that, but I mean, the issue I think is we need people to have the tools to work with the data that we're putting out there, right? And I say we're constantly modifying the setup of the data that we're presenting to people as the audience changes. That's the kind of dynamic aspect that I think we really need to think about. As you say, it started with the scientists, but you know, as more and more people find out about this kind of digitization work, the disconnect between what we're providing and the audience, what the audience needs, grows, I think. So we need to plan around that.
Just to not drag this on, but one comment of that is one specific way in which we've responded to people is that for those of you who interact with Ober, you'll know that at least at UF, it was possible to jump the scanning queue. Because if people asked that they needed this specimen or these data for some specific project, we usually just bump that up in the queue. Maybe it wasn't something we were even planning on doing, but I was like, if the whole point is to have people use it, what better way than to have people ask for it and then give it to them, right? And so I think, you know, that kind of over the course of Ober, we kind of evolved in that, or we were in fact sometimes responding to requests.
Other good questions? So far, we've had a lot of good questions.
This time, I was real quick question about the 360 videos. I was wondering if anyone's played around with stereoscopic 360 videos. So multi-camera from multi different, 360 multi-cameras that provide stereoscopic. Okay.
We track firefly swarm movements with 360 stereoscopic to understand collective behavior. It works really well to make 3D trajectories of these behaviors.
That gives you additional pinpoint accuracy to those things. That's great.
Yeah.
What system did you use?
Like what cameras?
Which camera? Yes.
Yeah, we just use GoPros, and
Okay.
we have citizen scientists that will send GoPros to also to collect data for us.
Okay.
And it works really well. It's great at tracking movement data, and because fireflies flash on and off, we can do it at night. But you do need light typically, but you can do it with like birds or other things too.
And use two.
Yeah, so you either put them 3 meters apart or 6 meters apart. We have people tell us how far apart they are. We put them in a, I didn't make the algorithm, but one of the people in my lab made an algorithm, and it'll spit out XYZ coordinates and make animations of the movement.
Thank you.
Nelson, you talked about focus stacking. It seemed, and I may be wrong about this, but does your COPPAS system sort of make focus stacking superfluous? I mean, you're taking images from all kinds of directions, so you must be getting some inherent focus stacking out of your method.
Well, so we still have to do focus stacking when we're limited by the optics that we have, right? So, you know, and it all depends on the environment. I mean, what's really nice is since it's so controlled, and we could, in essence, you know, leave the shutter open, wide open for 10 minutes and have a really long exposure of gaps. And so that could give us a really deep depth of field. But we try to, we try to do something that's more reasonable. And sometimes for insects, we do need focus stacking to get that high resolution.
Yeah, right.
So with the optics that we have, usually if we're shooting something that's smaller than 7 or 8 millimeters, we probably have focus stack.
Okay, we are at time. Next is going to be Zach Randall from the Florida Museum. Okay. Okay, we should be good to go. This is right here.
All right, some great talks so far. This has been really fun. So my name is Zach Randall. I manage the digital imaging division here at the museum. And today I'm going to be talking about fluid collection and photogrammetry. So for those of you not aware about fluid collections, these are also called wet collections in natural history museums. They are specimens preserved in ethanol, isopropanol, glycerin. And these comprise a huge amount of our natural history collections when we're thinking about fish collections, herpetology collections, huge amount of aquatic invertebrate collections. These are preserved in fluids. And to a lesser extent, you have smaller collections of ethanol or fluid-based mammals and birds. And here at the Florida Museum, we have around 4 million specimens preserved in fluids. So for us, trying to come up with a really good way to create photogrammetry models of our fluid collections is really important. So we do a lot of photogrammetry already of dry collections. Here are some examples of that. We just finished up a big project doing all freshwater mussels of North America. So we produced photogrammetry scans of 330 freshwater mussels in collaboration with U.S. Fish and Wildlife, the Smithsonian, and Chesapeake Bay Foundation. And the way that we do our high throughput photogrammetry is we use this designed 3-camera system paired with a turntable. So this is an automated system. Every time the cameras fire, the turntable turns or vice versa. And the way that we do the modeling is we'll do a whole 360 rotation of a specimen. And then after that 360 rotation is done, we'll flip the specimen, do a whole another 360 rotation, and then all of our imaging is complete for that. And this is what that looks like. So we have, this is in the reconstruction program. We use reality capture primarily. So we have these 3 circles. Those are our 3 cameras. And this is the dorsal or top view. And then we have the bottom view over here. And so the alignment program or the reconstruction program does a really good alignment with all this. All these tiles that you're seeing are the actual photos that were taken. And then we can go through the rest of the process that a lot of us are already familiar with for photogrammetry, doing the alignment and then meshing and then using the photos to create your texture. The problem is with fluid collections is that they are not solid objects. They have a lot of flexibility to them and they move a lot. So when we try to do the same approach and we try to flip our specimen between the 2 imaging sets, what you get is a lot of motion. So here the turtle head is shifting over on your left side. And this is problematic because when you don't have the consistency, the alignment stage and the reconstruction program has no idea how to mesh this object. And as a result, you get huge chunks for the misalignment that are missing in your meshing process. And it doesn't really work that great. We've also experimented some other techniques where you kind of hide portions of the object that might be more affected to movement, like this, like hiding in an ectofoam and then flipping it out. And for the most part, it actually kind of works. But then you do get these real issues with texturing. So part of the ectofoam was then used in the reconstruction program and mapped on the head. So while it works, it's not really efficient because there's a lot more post-processing that's involved to clean these models. So this is where this new system that I put together comes in. This is now in our fluid collection building, which you guys will be able to tour tomorrow. And I'm gonna just talk a little bit about it, but the main takeaway is that for this system, our objects are not flipped. So we avoid any of those chances where the object might move or shift during those two image orientations. Now it's just one image orientation. So this is with a 3-camera system. Because this is macro, we're using macro lenses on this. You didn't need a 4th camera. But overall, our new system is 4 cameras, so you get a nice options for angles from atop and below. And so the rotary table, instead of traditionally, it might be at the bottom on an actual table. Here we have it inverted, hanging from up top. We have our 4 cameras, and then our specimen is hanging from that. We use aluminum wire right now, and we're still experimenting with different attachment types, depending on the weight of the object that we're trying to model. And if folks have suggestions for this, love to chat more about it. And then we include our scale bar, which is done a lot for traditional photogrammetry, so that it is actually real scale to size. And here's an example of some of the models that we've created out of this. I will note that these frogs here were used with the macro setup with the 3 cameras. And I think we're getting really good detail. And one of the takeaways here is that the modeling is actually really fast for these with minimal post-processing. And the setup really lends itself to really oddly shaped specimens, especially really large things with long tails. So those you can't flip, right? If you flip those tails are going to move position. It's going to make reconstruction or alignment really hard. And then stuff for, we have a lot of fluid specimens that when you take them out of ethanol, their shape completely changes, right? So like this frogfish, you put it on a table, its belly is going to be totally flat, and then vice versa. When you flip it, it's going to be really hard to do this traditional flip on a rotary table setup. And the setup also lends itself to doing some other cool things like insect collections, which are pinned usually, and they can be hard to do on a tabletop setup. We have the capabilities now to do macro with the setup, getting really good resolution, coverage. Also doing somewhat of stacking, but we're not actually stacking all the images. We're more so just doing a couple of different orientations or rotations, I should say. And it also provides opportunity. Well, us here, we can't really see this fluid preserved rat, but it lends opportunity to start 3D modeling things that we wouldn't traditionally think of doing, like these fluid mammal collections or bird collections, and also lends itself to doing now specimens preserved in glycerin. So if we're interested in analyzing the differences between parts of an object that are ossified versus cartilaginous. So I'm just going to go over some of the system pros, some of the cons. I'm going to look this up real quick. All right, so the big pro is that you don't have to flip the specimen, which is great. Because it's a multi-camera system, imaging is way faster, which decreases your time of drying out these fluid preserved specimens, right? You have a fluid preserved specimen out of the jar, it's going to dry, which is problematic. Not only because it would damage the specimen, but also from a modeling point of view, parts of the object are going to start shrinking, and so the alignment is going to change. And also because we are positioning things now vertically rather than horizontally, you are getting much better coverage of the focal point. So for an example, with this 4-camera system, we've also been playing with what's the maximum threshold for the number of degrees that we can get away with while still retaining a good amount of resolution, both in our mesh and texture. And we found that we get really comparable results if we image every 8 degrees versus 5 degrees for the rotation. And this, with our 4-camera system, provides 180 images in around 3 minutes, which is really great for fluid collections. So here's just illustrating what I mean by a better point of focus. So this is our point of focus in the center. If we have a specimen on a table, especially elongate specimens, which are really problematic, your center of focus is going to be more in the center. And then if the distal and proximal ends of the specimen, they can easily go out of that focal range, where when you set the specimen up vertically, it's all going to be in that more so centralized point of focus, which is really great for creating models of elongate specimens.
Time check.
Time check, okay, almost done. All right, so cons, right? All these setups have particular cons. What are ours? So snakes are very problematic so far. We're still thinking about ways to solve for this. I think we do have a way to do it. But also really want to talk with folks, hopefully tomorrow, about some ideas for this. Glare, imaging anything out of fluid, glare is going to be an issue. And then in general, fish fins are really hard to model because they are so thin and they're usually transparent, which photogrammetry does not do a great job with. All right, so for glare, I think we solved this issue. We just use polarizer filters. Sometimes we do cross polarization where we put polarizer filters on our light. And this takes out all of that reflection. Here is an example of what that looks like. On the leaf, you don't have polarizer there, you get all that shine. And then when you put the polarizers on, it gets rid of that. So that's been really great, check for that. So that's not really a big issue, but the really big issue is fish fins so far. We do have a way to fix this, but I don't think it's a really long-term efficient way to do this because we do want to do mass production of these models. But currently what we're doing is we'll run the object in the software, fix the fins in Blender, all this stuff, and then rerun it through the software again, where it reapplies the texture on the fixed mesh. So also really would like to discuss with colleagues here on some potential ideas of how to get better coverage of the specimen and hopefully maybe fix some of these issues with capturing the fins. So maybe check, not really. So last but not least, I just want to give a shout out to the fluid collection imaging crew here. They've done a great job. And they have been producing a lot of these fluid collection models that you would see on our Sketchpad page, which please check out. Here's the QR code, and special thanks to funding sources, the Florida Museum, IDIG Bio, NSF, and the U.S. Fish and Wildlife Service. So thank you very much.
Adams. So Adam Roundtree will be up next from the University of Michigan. Yes, it should be in PowerPoint. I think you're sharing your screen, so you're good.
Already sharing?
Oh, nope, just kidding. You do have to share your screen again. Sorry.
There it is. Okay, cool.
Hold on, I think sorry.
Yep.
All right, so I'm Adam Roundtree from University of Michigan Museum of Paleontology. We're going to be talking about our sort of production level methods for fast, consistent, all sides models for rigid things, not for wet collections. Okay, so just going over some of our output goals. We want one model that includes all visible surfaces. So that's the all sides aspect. We want it to look like it's floating in space like that. We want 2 linear measurements on our model to be within 150 microns of 2 linear measurements on our physical object. We want consistent color information from one model to the next. Color information that's at the scale of the geometric information. So we're going to use vertex colors instead of texture maps to avoid having models that look like they have more detail than they really do. And then we want a watertight mesh so we can do 3D printing and all sorts of other processing. In terms of the goals for the method, so we want to cover a wide range of rigid objects without requiring a bunch of technician experience. So undergrads do 99% of our imaging, and so we want the model that freshman who comes in the first day of the job, the one they produce will be the same as one produced by someone who's been doing it for 3 or 4 years. We want it to be fast, obviously. We don't want to have to do any manual operations like masking or control points for alignment. We want the same lighting conditions for everything, same camera settings for everything, and minimal post-processing. So how do we get there? Starts with our photography space. So we're trying to get these all sides models. We're going to flip the model halfway through. So we need really diffuse lighting coming from all directions so that we don't get any strong directional shadows or any dark areas on the final model. So we have this large diffuse lighting box. We just made it out of foam core, so like $10. We shoot 400-watt LEDs through that. There's a tripod for the camera, obviously. And then we have a bunch of different turntables that we use depending on the size of the object. Two of those involve an Arduino control that triggers the camera and rotates the object. And then the most important part of the photography setup is the acrylic ice cube. So you can see down in the corner for this Mosasaur radius, it's sitting up on these acrylic ice cubes. And what that does for us is it allows light to come from beneath the specimen. So we don't get dark shadows where it's in contact with the turntable. So they're transparent, obviously, but they're also wavy on the sides. And so they refract light in really weird ways. They're completely invisible to photogrammetry algorithms because of the way they reflect light. So it's like our object is floating in space, basically. Okay, for the camera, we're using a Nikon D810 with a 60mm macro lens. ISO is set at 64, that's the lowest real ISO of the camera. White balance is just fixed at 5600 Kelvin, which is the color temperature of our lights. Shutter speed ends up being around half a second, and that's because we're shooting at f32. So yes, we are getting diffraction blurring at f32. But through some tests, we found that increased depth of field is more important than hitting like peak sharpness for the lens at f11. So if you look at the two different images here, the one on top is the f32 and f11 on the bottom. So even though this part of the object might be slightly sharper in the f11, we get longer track lengths with f32. So those points are tracking through more photos, which helps with alignment and sort of the final quality of the model. So that's why we're shooting that aperture. The process really similar to what other people are talking about. So we set things up on the turntable. We have 3 angles. We flip it over. We do 3 angles again. Most of the students like using the Arduino setup, so they don't have to move the turntable. And they usually take about 25 photos per position, per camera position. So we end up with 150 photos. Alignment time for the cameras there is about 1.5 minutes. We're using reality capture like everyone else, which by the way, is changing their license agreements in April. So we should start to see real educational use without a bunch of terms and conditions beginning pretty soon. Then our final output there, we get a mesh. It's 19 million faces in this case. We colorize it using vertex color, and that total processing is 6 minutes. We do our scaling after the fact in MeshLab. So we take measurement on the physical object and a measurement on the digital one, calculate a scaling factor, stick that in a sidecar, scale it up, and then check again our 2 measurements to make sure we were within 150 microns. And that's done for every object. We're presenting these online on this repository of fossils website. So we're going to need to reduce the resolution of these from 20 or 90 million or whatever they are down to about 2 million faces. We do that in MeshLab. We keep PLI files for the sort of archival copies, and the ones we're serving through the interactive viewer in CTM format. So we use the open CTM software. And one of the reasons for that is because the default MG1 method in that uses LZMA compression, so it's lossless. Our floating point data in the compressed model that you see online is identical to that in the original model. And those files are at like 8 megabytes. So we make these available on our UMorph website, which I've talked about before. That 2 million face limit has been in place since 2014, and it was based on the model working on a 2010 smartphone. So we're trying to reach a broad audience. with these models. And I'm not going to talk about it a lot here, but everything on the website is covered by our terms of use, which, because these are natural history objects, we're not talking about copyright. This is basically a contract that mimics a Creative Commons attribution, non-commercial license with a few little extra pieces for covering distribution and 3D printing. If you're interested in licensing, chapter 5 of this book, the CS3DP book, covers licensing for 3D models, gives a bunch of examples, and there's an open access version of this at that URL. Okay, so I was also asked to talk about RTI if I had time, which I guess I do.
Yes, you have 3 minutes.
Sweet. So RTI, reflectance transformation imaging, this is something that we use for flat objects or almost flat, like fossil fishes and fossil plants. It's been around for about 25 years, previously referred to as polynomial texture mapping or PTM. It's used a lot in archaeology. It's basically taking photos with a bunch of different lighting angles and then incorporating those into a single file that you can then relight or calculate surface normals from. So like this clay tablet allows you to make it have this metallic look so you can see what's engraved on the clay tablet better. Typically this was done with lighting domes like this, where you've got fixed positions of LEDs within the dome and there's a computer controlling the light during the process. Or you can do a sort of more manual method with strings and flashes. We have all these turntables, so we thought, well, let's see if we can do something a little crazy and stick a copy stand on top of a turntable. So we put the turntable is underneath here, and the lighting is just fixed next to it. You can see the little fiber light in the corner. So we just rotate the entire thing, take a bunch of pictures as we go. We'd be stopping it to take the photos. That's just to show the rotation. And the reason why this works without knowing the exact position of the lighting relative to the specimen the whole time is because we're using these black marbles in the photos. So this software, Relight Lab, is able to calculate lighting angles if you have spheres in your photos. So we use black marbles, but you can use ball bearings. The black marbles seem to work a little better. So you just tell it where those are. It finds the highlight, it does that through all your photos and then produces an RTI output, which you can then view in RTI Viewer, so you can make it look metallic like this. You can also show the surface normals. That's a desktop program. There is a web-based viewer that we use on the UMorph website. It doesn't give you quite as many options for relighting, but it is interactive. You use tiled images, and you can relight the specimen online. So this is a really good way for us to share things like our fossil plants and fossil fishes, where a 3D model wouldn't have maybe enough resolution to show the features in the surface. Right. And that's it. So there are a bunch of links for the software that we're using and other resources.
All right, next is Alex Atkinson from Florida State University.
A couple videos on the video.
Okay,
Here.
thank you.
Okay, hey, how's it going? My name is Alex Atkinson. I'm here at FSU through Austin, Mass, and collaborating on this with Barbara Costa and Alex Leeds, who's doing a lot of the kind of back-end development on some of this stuff. So I'm focusing on 3D Gaussian splatting and neural radiance fields and surface reconstruction from those algorithms. And it's building off of previous research into photogrammetry where we were trying to capture specimen in the field. And we found that you can efficiently do that, but it kind of caps out at certain levels of complexity per specimen, like these delphinium here. You can see there's kind of some data loss or like additional kind of warping, as we've seen with photogrammetry. I'm sure you're very familiar with this. And then specimen like that dandelion on top are just like kind of altogether impossible due to the nature of photogrammetry. But with these new algorithms.
Yeah, did you see,
Oh,
it
shoot.
says, yeah.
Two-factor.
We've all been there.
All right. So as you can see, these algorithms are able to accurately reconstruct even these extremely fine filaments of a dandelion. So kind of an exciting development there. Perhaps it could resurrect dead data sets because of this.
I think it's click the top tab. Up in the browser. Yep, there you go. Oh, no,
No,
that's
hang on.
not in the right place. Yep.
There we go. Okay. Yeah, so what's happening here? It's able to take missing data that can be inferred through sort of like secondary sources. So you're taking photographic input, RGB, it can be video as well, and then it's running it through traditional computer graphics algorithms like ray tracing and sort of these other typically non-photo reconstruction algorithms, and then applying those with neural networks to photographs to be able to reconstruct lighting scenes and information from that by pixel density or surface information of objects or scenes. Okay, so here's a breakdown of the pipeline that we've been running so far. All of this is built on photogrammetric architecture, which is really nice. It's all open source. Very emergent, and a lot of sharing of these algorithms and a bunch of different techniques. So this is just one possible pipeline. So you take kind of that first step of matching and feature extraction from photogrammetry, and then you're applying that to then either the neural radiance field or 3D Gaussian splatting algorithms, which take up those structure for motion points and then run that through a different rasterization process, either the neural radiance field or the Gaussians. And then run that through a neural network, which is refitting the lighting information, pixel density, depth of field, things like that, from back to the images and continuously refitting that. And then it does a very similar process to also gain a mesh that you can extract from these. And again, there's many kind of interoperable pipelines where you could use various algorithms within that. Obviously, everybody seems pretty familiar with photogrammetry, so I guess I won't really backtrack on that too much, but this is the data, the first kind of step that you're taking, those camera positions, and then your sparse cloud information. And then what happens, so this is where things get a little, thank you, a little bit different. So in the case of, on this one here, we've got, this is the neural radiance field. So you can see it's then extrapolating out many projected rays of light and essentially kind of bouncing them off of the objects or other things in the scene, the entire data of the scene, and then refitting that again against those images. The 3D Gaussian method is a little bit different. It's fitting an array of these Gaussian distributions of pixels. So they've got pixel density and color data on that Gaussian distribution, and they kind of stack those in like harmonic spheres and then map those to a depth map. Maybe, yeah, there we go, cool. So here's, yeah, just a further breakdown of what that looks like. You rasterize, you go through the neural network, it fits it to the images and then recreates a scene based on that. And here's just a couple examples of the differences. So this one here is, these are pretty low resolution, so kind of to emphasize some of the information in the scene. This is a neural radiance field. You can see it looks very sort of pixelized. These are actually voxels, so they're 3D pixels, a little bit more data there. And these are all running on my 4060 Ti graphics card, which has 16 gigabytes of RAM. This is a Gaussian splat, and you can see that it's kind of hard to tell here from this low res, but there is a little bit more information and accuracy from the Gaussian method, and it tends to be somewhat significantly more computationally efficient. So that is why we kind of decided to focus on that side of things. And then we were also able to find out that you're able to get decently accurate. We need to further test this, but so far we're down to about 100th of a millimeter across different points of a mesh that's been extracted from that. And this is, I think, maybe a pretty exciting moment. So this is a head-to-head comparison against photogrammetry using Metashape. Exact same data set, untextured mesh here, using downsampled images to 720 pixels. And you can see that there's a lot more detail on that mesh from the exact same data set. This moment right here, this kind of broken occipital arch, is something that, I wanted to make sure was kind of in the data set. It was kind of our focal point. And so here's a 6K resolution image from a mirrorless DSLR. And here's an untextured Metashape mesh from those 6K images. So this is like very high res. And you can see it still fuses that arch. So it's kind of exciting that we're able to get on the 720 data set, it's able to maintain that geometric accuracy. Here is a hybrid render. These are kind of like real-time renders. So they're sort of weird. The texture is applied through real-time rendering. So it's a little bit different method, but that's what that looks like. This is, again, downsampled to 2K because now we're using Amazon Workstation Cloud GPU, and we already capped out our 24 gigabyte VRAM at 2K. So that's what that looks like. And there's a lot of headroom there for that, using better GPU systems and things like that. So also kind of exciting. There's a 6K Metashape fully textured mesh. So as we've seen, photogrammetry, especially on optics like this, can produce amazing results. But that's the head-to-head against our 2K render. So getting pretty comparable even on the textured mesh. And again, maybe maintaining some better geometric accuracy. And here's just what that full, here's what that full render looks like in the real-time render view. Pretty comparable to photogrammetry, even at those lower resolution scales. So, okay, so next steps, we want to kind of test this up against things more traditional measurement methods like a microscribe perhaps. We're hoping that this opens the door to interspecies study, like amongst a population where you might need a lot of data sets to have this opens the door to sort of more rapid capture. As you can see, you can have a more incomplete data set and lower resolution images. So it's a lot more efficient and can use more incomplete data sets or fewer images to get, in theory, a higher quality reconstruction of that model. We want to look into how far we can push that kind of next. And just a few other takeaways from these methods is they're really good at capturing like surface reflectance because they are like light-based reconstruction methods. And these extremely fine, like the filaments of this azalea here, that image or that model was captured in the field. That flower was moving around. That was my cell phone captured that, and it was still able to get those filaments. So again, it really kind of opens the door to some other conditions of being able to model out in the field with like pretty decent accuracy, even in much less than ideal conditions. Again, it's very, very efficient in terms of the data sets. You do have to kind of outsource then your upfront costs of maybe like doing all these really elaborate photogrammetry methods. You do have to have a really nice GPU, but the capture method might be a lot more efficient. It could potentially resurrect some dead data sets because it's able to get things out of it that the previous methods weren't. Again, that unconstrained capture, and it is so far we've driven down to 100th of a millimeter accuracy. Yeah, just some acknowledgements there. Thank you.
Now know how we're gonna do fish guns.
Yeah, right.
Okay, and yours out there, hold on, sorry.
You have to download this. I will edit out.
Right, Arthur Porter, Florida Museum, thank you.
All right, let's see. Very good. Thank you all very much. Thanks IDIG-Bio folks for the invitation to take part on this. So today I'm gonna talk what's a very, and I can't emphasize this enough, a very recent interest of mine. And I wanted to talk a little bit about some work I'm doing with regards to color. But why color? As you all know, and I don't have to spend too much time on this, color is a critical component of organisms, right? Like it plays a wide variety of functional roles within species, both in terms of signaling, camouflage, mimicry, and even temperature regulation. But despite this being such a critical component of organism morphology, color is something for which we have very little consensus on how to measure and compare across species. So especially when you compare with size and shape traits, where we have 3D geometric morphometrics as a tool set that anyone can use in any context, the tools that we have for color approach the problem in very different ways, and there's very little consensus on how we actually should do that. And even more important than that, they're almost always restricted to the 2D realm. So we really don't have the ability to process 3D color information or a good tool set to process that information effectively. So some of this is natural, right? We know that dealing with color can be a lot more complex than just size and shape, in part because there's a lot of sources of variation. So there's sources from biology, like if you have mussel shells and they all have different colors, but they also have different shapes. So how do you do the mapping from this shell to this shell is not a trivial aspect. The other thing is that most images that we capture are to a large extent and standardized. So you get situations like this where you have two pictures of the same specimen, one under diffuse light and under direct light. And as you can see, they look remarkably different in terms of their RGB values. And finally, there are things like UV, right? So when you take an image of these two species of the same genus of butterfly, under traditional light, they look fairly similar in their coloration patterns, but then you throw them under the UV light, the second half, and they look completely different. So it's natural that color is harder to deal with, and there is less standardization of how we approach this. But even then, I was surprised when a few weeks back, Zach and Laura arrived in my office and they asked me like a simple question. They were like, look, we have this photogrammetry model of sclera ossicle from Mahi-Mahi, and they wanted to do a simple quantification, right? They wanted to know what percentage of the area was bone, what percentage was cartilage. And my intuition, when they first approached me, was like, oh, okay, this is gonna be simple. That would be something that we can use to just, you throw that into a software and it's gonna spit out like an area that has kind of reddish wine color and the area that is blue. But
we were surprised to find they actually don't have those tools. Those tools simply don't exist. So spent some time and created a slicer module that basically does that. So it's tentatively titled Biomodel Colors. And the module basically requires two inputs, right? You need a 3D model of your object and the texture file, and then it has a very simple pipeline. So it starts essentially by wrapping the texture around the 3D model. So you have the texture in 2D and the 3D model wraps it around. So and it actually maps the color information through each vertex of the mesh. So this is each point in the mesh has an RGB value associated with it. From then, you can perform any kind of clustering method. We opted for K-means clustering with 2 clusters, right? Because in this case, we wanted to separate that into a binary model where it was either bone or cartilage. So you got this binary model in which every vertex color gets replaced by the centroid of the cluster. And once you have that, you can just split your mesh into 2 pieces and segment your 3D model and calculate some kind of measurement of what percentage of that mesh is bone, what percentage of the mesh is cartilage. So that worked well for the kind of thing that they were aiming for. But ultimately, it's something that is like not that satisfying, right? Because we work in biology and biology is comparative. So how do you go from an approach that works well for a single structure and that does the job that you aim to one that works with another one of Zach's amazing data sets with the freshwater mussels? So how do you go from one to the other? So today I just wanted to briefly present a proposal for myself of how we might approach that problem. So ultimately, I think we will only be able to deal with color information and find a tool set that can deal with that information in 3D if we incorporate that color, our color tools to our 3D morphometric tools. And essentially because morphometric tools are basically tools that aim at doing one thing, which is to place specimens in a shared coordinate system. That's what 3D morphometrics does. It places individuals in a population in the same coordinate system so that we can quantify and compare these things across individuals. So in my view, we're only going to be able to deal with color effectively and create tools that quantify and compare color effectively if we place those individuals in a shared coordinate system in which you can say this point in this individual is equivalent to this point in this individual, and you can therefore use traditional quantification, principal component analysis, whatever you want to do with that kind of data set. And really those tools exist. Pieces of those tools exist. So just an example pipeline that one could use that I just applied to the freshwater mussels that Zach has collected. You can start with your samples. In this case, the 3D models are samples. You can collect semi-landmarks in these in kind of a grid fashion, and they don't even need to be in homologous positions or anything. So then you can use landmark transfer tools such as Alpaca, where you use deformable registration to map one 3D model into another. And then you can get these sparse homologies, so to speak, into dense homologies, where with tools like DECA by Sarah Roth, where you then basically establish point-to-point homology at every point in every 3D model to every point in another 3D model. So essentially, each individual in the population then becomes a deformation of a mean mesh and associated statistical shape model that describes the variation in the population of that mean mesh. So essentially, each individual in the population then becomes just a deformation of that mean shape. And with that, you can then deform all specimens that you started with into the mean shape of the population, and therefore created this shared coordinate system in which color information is mapped into. All right. With that, then you can then go and use more traditional color tools and then perform, for example, you can perform an initial color clustering, so you reduce the amount of noise that is associated with lighting and all that kind of sources of variation in color information. Obtain color maps for individuals. You can reduce those color maps using clustering techniques so that you merge colors that are too similar. And then you can merge those color maps together again by placing all color maps of the different individuals in the population, doing hierarchical clustering of those colors. Again, creating a threshold where you merge colors together. And in the end, you can impose this shared color space at every single individual in the population. So then you end up with individuals that are not only in the same coordinate space in terms of spatial traits, but also in the same space of colors, so to speak. And when you're at that situation, then you can just, it's data that can be just played with. And you can do things like, for example, do a principal component analysis of the color information across individuals, find clusterings in that space, even among individuals that if you look back at the slides, they're completely different in shape. But when you actually place them in these shared coordinate systems, you see that their colors are all, they map to the same region of that space. And you can calculate similarity between individuals, find individuals like this one. In this case, this is a 2D plot, but if you actually had more dimension on this plot, this individual would fall completely outside of the rest. And which is this individual here, which is not similar to anything, and it lies at its own by itself. And with that, we finally perhaps can have a quantitative framework where we can analyze color in the same way that we analyze size and shape traits. And that's what I had for today, and I want to thank a bunch of folks that helped in that process. Thank you.
So we have, it's time for question and answer again. Austin Matz is going to help lead us for this question and answer. So if you have, I'm going to stop sharing. I'm going to turn this around. So hopefully everybody out there can see everybody here better. And if you are online, please add your questions in the Q&A or feel free to raise your hand.
Ed has a question.
Yeah, I would ask a question for Arthur. So with the warping of the data transfer, that doesn't impact the relative size of the individual triangles because you move it back to the original shape, right? I was thinking about the proportional makeup of different colors. So when you're warping the specimen, warping mesh, does that modify the relative proportions of those meshes? Or?
Yeah, it does. Yeah. You can deform like things to the mean shape. Then the size of the triangles relative to the initial won't be the same.
Will that impact the kind of the makeup of the proportions of colors? If the kind of the middle is warping, then that seems to be the main part.
If you have very low resolution, you couldn't get those color patches correctly. Yeah, probably. But if you have enough resolution in the vertex, because essentially we're mapping back to the vertex, right? Each vertex. So as long as you have enough resolution, that I don't expect it to be. But if you have a very low resolution model and you're trying to do that, then perhaps you get a larger spectrum.
Other questions? Are there any online?
I have another question. I've always got more questions about the photographic stuff. So transparency, Zach, you mentioned, the fin rate thing. That's always going to be a challenge, right? How do folks who work with the photogrammetry work handle transparencies or translucent material?
I mean, one way that we solve it is reconstructing that portion of the mesh using and running, because the textures and how it's really just the meshing part of the software that just can't really do aspects. But once you create that mesh in that area, you can still then utilize the texture from that exact region we plotted. So that's my experience with it, is really you just have to go in, you can do some post processing in Blender, fix the mesh of that area. Is that?
So there, this would be probably difficult to do with specimens, but there are sprays that you can spray on the material and the stuff can make away. And then you can retexture it out. So you would then be building your model against a kind of like covered specimen. And then after a few minutes, when that just appears, you then use those. But I don't know anyone's ever done that, for specimens.
Yeah.
At least I feel like graphic arts.
I guess fossils, something, you got a shiny fossil.
Yeah, you could do it to also cover up shiny.
That wouldn't work too well regularly. Flexible documents.
Probably not. And the fact that it's a spray and,
So, I've not tried this, but just thinking of that specimen. Have you tried imaging them in infrared? They might not be transparent. It's worth a shot.
Yeah, that's a good idea. One of the issues is, is transparency, but also how thin they are. And that when something does start to dry, it's usually starting at the ends first, and then that's when you start getting the shape differences and the alignments are just off. And that's where you get, that shape that's in line too, but that's definitely, right, that's a good idea. Good.
So from online, what methods can be used to ensure color capture information is standardized to an accurate color value beyond ensuring all imaging are taken under the exact same lighting conditions?
So it was mentioned before using color checker during your imaging and having that in the beginning. What we like to do is not actually post-process the white balance of the color correction when you transfer all your images in, just because it takes along, it's another step and takes time to do that. But we more so just use the color checker as a calibration on our actual texture file that's later produced. And, there has been a lot of conversation actually recently in a listserv talking about the accuracy of color in general for 3D models, especially in photogrammetry and light scanners, that there are problems even with how the texture is created, that it's not always as accurate as we want it to be. So it's still developing.
I have my own question. So like when we do like remote sensing field stuff, we typically have like algorithms that try to at least correct for how much light is coming into a sensor. Is anything like that being done, like to help standardize like lighting across stuff? Like images for color and stuff, or is it pretty standard, like how much lighting is being used for taking pictures of specimens?
We have like our standard light setup we use to use lighting, flashes, and then I think for the standard, you could look at whatever your aperture is set at. Yes, if you want to use that information for metadata to kind of understand how much light is coming through. Is that kind of the question?
Well, kind of the question is like, how do you standardize across collections if you're pulling data from multiple photogrammetry collections that you want to use for training data? Like, how do you standardize across multiple collections?
You want to go through?
I was just kind of gonna build off that question as I've mentioned everybody talk about color and approximating color, but I haven't mentioned anyone talk about color spaces. So in photography, there are specific color spaces that most photographers and museum collections use, like ProPhoto. A big one is Adobe RGB, and they're these big color gamuts of spaces, but I haven't heard anyone here mention them. So like you can actually use them in the program, but also in most of the digital programs, you apply this profile, and this profile can then be kind of collaborated or then applied to your workflow, and it how to kind of answer that, you use the same color space to recognize those colors. But then people talk about approximating a 2D color space to a 3D application where you can basically create your own colors based off of the original color space provided. But there's certain gamut ranges that could create false color, but there are certain spaces that, depending on if you apply that and make an overlay, you can actually create an overlay of a color space that exists and then apply it based off of kind of what Zach was talking about with real color or approximations of color using laser shading and photogrammetry, but you guys kind of talked about that.
All right, well, we should earmark that for later discussion or maybe lunch discussion. We're going to move on now to the next talk and
Should be
Jill.
ready to go. Turn the camera. Yes, thank you. All right, Jamie Gray from the Florida Museum.
I tried to get dim the lights a little. I would
Exactly.
Everything just looks better.
Yeah.
Good. No, that's good. Thank you. Okay, yes, I'm Jamie. I work here at the Florida Museum in the Digital Imaging Division with Ed and Zach. And I work on the OVERT project. So by now we all kind of know what that is. If you didn't see Dave's talk earlier, there's a little bit of info on the screen for you right now. But what I really want to talk about is the contrast enhanced CT scanning portion or soft tissue scanning portion of the OVERT project, because that was what I was hired to do, and that's what I spent the past few years on. So the way that I did this was by using diffusible iodine-based contrast enhanced CT or DICT. And this is a way of using iodine to increase the radio density of soft tissues in CT scans. So let's say, for example, I'm interested in making a 3D model of the brain of one of my favorite animals, a highly venomous eastern brown snake from my homeland. I get a museum specimen and I submerge it in a solution of iodine. And then I wait long enough to allow that iodine to diffuse into all of the tissues and saturate them all. And then I pop my snake in a CT scanner. I CT scan it, and now the iodine has bound to the carbohydrates and lipids that are naturally present in our natural history specimens and increase the radio densities of all of those soft tissues so that I can make a 3D model of my snake's brain. Then afterwards, I can actually destain or leach out the iodine and pop the specimen back on the shelf where it can be used for other things in the future. So here's 4 examples of some of the couple of hundred data sets I've generated over the past few years, a fish, a mammal, an amphibian, and a reptile. So on the screen, you see a single cross section from each data set. Each data set is made up of about 2,000 of these, all the way through the entire specimen. And just from this snapshot, you can get an idea of the kinds of structures that we can isolate and study using our DICT scans. All of these are available to download already on Morphosource. So there are some QR codes on the screen. There are separate projects each for fish, mammals, amphibians, and reptiles. So pick your poison, peruse at your leisure. I'll pop these back up on the screen later. So I learned a lot through doing this. I DICT'd over 200 things, and I didn't actually write a diffusion diary, but if I did, this is what it might include. Some of the challenges presented by trying to DICT museum specimens, the predictions of staining times for different kinds of specimens, and some of the hidden secrets that are harbored inside of our natural history collections. So let's begin at the first chapter, challenges. Dear diffusion diary, it's been a few years now, and I'm going to tell you there are some challenges when it comes to working with museum specimens. Perhaps the most major one of these is that I am a digital imaging specialist. I don't have any control over how these specimens are collected or fixed or stored. So the best thing that I can do is try to choose my DICT subjects wisely. One of the most important things that I consider when I'm choosing what to DICT is the preservation history. The general recommendation for DICT has always been that fresh is best. The best thing you can do is euthanize your animal in the lab, fix it immediately, and then image it afterwards. It's also recommended that you aim for the youngest possible specimens. It has been suggested that long storage times in ethanol can lead to degradation of the soft tissues. I don't have control over this part, so most of the things that I'm working with are things that have been fixed with formalin and then stored in 70% ethanol for decades. But I've still gotten some really great results. So I want to show you a couple of examples. This is a little horned toad from the Yale Peabody Museum. It was collected in the year 2000. So it was stored in ethanol for 23 years before I imaged it. And here's a cross section showing you that the tissues are still in really nice condition. This is a common bigmouth toad, also from the Yale Peabody Museum. This specimen was collected in 1929, so it had been stored for 94 years before I got my hands on it.
I imaged it and the tissues still look really nice. And in fact, this is one data set that I typically use as an example of really nicely preserved tissues. But don't just take my word and those two specimens for it. Let's look at some data. So I came up with this, thank you, a tissue condition scoring system from 1 to 4, with 1 being very poor quality and 4 being very good quality. This is a scoring system that basically reflects the condition of the tissues and the usability of the data sets. So I scored 204 full body scans of specimens from 11 different natural history collections. And from the tissue condition scores, we can see that most specimens resulted in good quality data sets, with 85% of them scoring a 3 or a 4, and 15% of them scoring a 1 or a 2. Now, going back to the recommendation that you should choose young specimens, I have enough data here to actually see that this was true. They were collected from the year 1924 all the way to 2022 or 23, so almost spanning 100 years. And when you bin these out by decade, so on the x-axis you see the year of collection binned by decade, on the y-axis you see the number of specimens and they're colored by their tissue condition scores, we see that it doesn't really seem to matter when a specimen was collected. What matters is how it was treated upon collection. There are things that have been stored for decades that still image really nicely. Okay, moving on to the next chapter, our predictions. Dear Diffusion Diary, amphibians, fish, mammals, and reptiles all behave differently when bathed in iodine solution. So throughout this process, I collected body dimension measurements and staining durations for all of the specimens. And you can see here a log transform plot with weight on the x-axis and staining time on the y-axis. In general, the bigger you are, the longer it takes to stain you. And we can see that fish and mammals of a given size typically take longer to stain than amphibians and reptiles of a given size. And that mammals also have this very steep increase in staining time, perhaps due to their overall more bulky body sizes in comparison with some of the more streamlined bodies that were in this data set. Using this data, I came up with some predictive equations that will allow you to predict how long staining might take for an animal. There's a lot of variation in the data set, so this estimated staining time should be considered a starting point, and you should know that your end stain time may be very different depending on what structures are present. Also, this is for the 1.25%, which is a low concentration of Lugol's. So if you predict your staining time and it seems really long, you can consider increasing the concentration of your iodine, and this will speed up the rate of diffusion and staining will occur more rapidly. Okay, so now for the final and my favorite chapter of the Diffusion Diary, the hidden secrets. Dear Diffusion Diary, many museum specimens harbour hidden natural history records, and these can tell us important information about the life history of these animals. So I just want to show you a couple of examples of these. Starting with our yellow-throated plated lizard or our pterosaur, this guy girl took a really, really long time to stain, because she is heavily armored, but also because she has 4 very lipid dense eggs inside. And in fact, you can see they're not quite completely stained yet. We also found some food items in the mouth, which were rendered in 3D, and they turned out to be seeds. And we found these seeds in much the same condition in the hindgut of the lizard, which tells me that this lizard had a hard time digesting this food item. We also have a fish, a lined lantern shark. When I scanned this fish, I was looking through the tomograms and I saw something a little strange. And when rendered in 3D, you can see that this is actually a squid or pieces of a squid. After consulting with some cephalopod experts, we put the identification down to the family Anaploteuthidea, likely belonging to the genus Abralia. Then lastly, we have our Texas horned lizard. I could tell just by looking at this thing that it had a very round belly. Inside, I found some eggs or some follicles, so this tells me this individual is a female. I found some parasites. I found lots and lots and lots of parasites in this thing. And in fact, there were 2 different kinds of worms in the stomach and the gut of this lizard. Also in the stomach, lots of food items, lots of ants and other arthropods inside. So all of these challenges, predictions, and hidden secrets became the inspiration for a new resource that I created, which is a digital guide to DCT
This is an interactive PDF. If you click through it, there is about 2 hours worth of total content. For anyone who's wanting to DICT their own natural history specimens, I'm happy to email this to people if they would like a copy. And I want to thank all of the co-authors and collaborators, all of the institutions that let us put things in iodine, all of the funding sources, and I'll just leave these here for a few seconds so you can scan the QR codes if you would like. That's it.
Ed's next, yeah.
I think the last time I gave a talk here was right after the election in 2016, so I hope the audience is a little bit more responding than it was then. Just a lot of people looking at the ground. All right, is this the right thing?
Yes, just do the share screen, Zach.
We're on, yeah?
No, once you go, it's gonna go back to,
You're sharing with me.
yeah, but you're sharing from PowerPoint, not the, I have to click it.
Is this good? Okay, perfect. All right, so you've already seen the screen. So I'm going to talk a little bit about I guess some of our takeaways from the OVERT project, which just finished, and Jamie talked a lot about this section here. But I'm going to really talk more about this and some of the things that you think would be obvious, and some of the things that are probably less obvious. So the museum, the OVERT project just finished, and we have scanned what, 13,000 specimens, and almost double that in terms of the amount of media files that we've put up online. It's been a huge effort, and I would say that the data going up, I hope, will have a huge impact on not just research, but education and artists, and all the things that Dave was talking about this morning. But I think for us as the digitization community, the takeaway, the thing that will hopefully have the most impact for us going forward is the OVERT pipeline that we've been building and working with museum collections and data aggregators like IDBio and the repository like Morphosource and Sketchfab to integrate this and build a fully integrated pipeline that funnels data into the public sphere. So we just had the paper come out. There's been a lot of press, and the journalists that do call me always ask me, so what's next? And the obvious answer, of course, is that, well, the OVERT project focused on neontological vertebrates. And so there's all of paleontology and lots of non-vertebrate neontological collections, like entomological collections or botanical collections, and also, 2 million other vertebrates that are sitting in collections around the world. So the OVERT project doesn't end, and the digitization, the CT digitization doesn't end with OVERT. And so we can use this pipeline to do entomological project, and you can CT scan insects just as well as you can CT scan vertebrates. Just requires a little bit more, I guess, higher resolution. We chose Morphosource as our repository because it intersects very well with museum data. In fact, it was designed to be used with museum specimens, right? So every single media file is pinned to a specimen, and that specimen interacts with or is linked to specimen records and specified through IDBio. So that's a useful tool, and I really want to kind of just focus on this portion of the work because this is the portion I think is a little bit of the bottleneck at the moment. We'll talk about the bottlenecks going forward, but the scanning of the data is not necessarily the hardest thing anymore. I feel like the part that people really lose steam with is getting that data into the repositories. So Morphosource is, you have to sign up for Morphosource, of course, but it's a free resource. Once you sign up for it, you have to register as a contributor. So once you get contributor status, you'll notice some contributor tools in your dashboard here. One of which being batch submission. And that really does speed up a lot of the process. So we can actually upload large amounts of data, multiple files, all in one go. For those of you who haven't used it. So once you've clicked on that, you'll see this batch submission form, and you'll see that there's a series of basic details you have to enter about the group of media that you're about to upload. Ultimately, the individual metadata is filled out by using this Excel spreadsheet. So because you're uploading a lot of things in one go, thank you very much. The data needs to be from the same collection and the same collection in the same institution. It needs to be managed by the same manager or group of people. It needs to be imaged at the same institution on the same machine, right? These are all information. These are all information that is shared between all the media data that you upload in one go. The ownership and permissions remain consistent across the batch that you upload, so that has to be all the same. And if you wanted to assign it to a project, that's a grouping of media that isn't tied to a collection or a data management system. You have to upload it there. So then you upload all of your data files via SFTP, FileZilla, or CyberDuck to a server at Howard Duke, and then you build this batch manifest, which is an Excel spreadsheet, which has 50 fields. So 50 fields of the different data, right? So when we were doing this the first time, we thought it's not actually saving us a huge amount of time because we're just cutting and pasting out of the metadata files into this Excel spreadsheet. This is what this looks like. So you have, the media file here, the description of what kind of data it is, all of the, your IDBio specimen metadata, you have to fill that in as well. So it's actually time consuming as well. It doesn't save you a huge amount of time. So we built because I don't know how to use Python, I built an R script to basically scrape your metadata from either Phoenix systems, Bruker systems, or Nikon systems. So if you can gather all of your metadata files into a folder, you can run this R script, and it will build that batch manifest for you, which should save you a bunch of time. There's some things you need to standardize, like the metadata file names all need to be standardized and include the Darwin core triplet. And that allows you to pull down all the data from IDBio. The delimiters have to be consistent, and if you want to include a descriptor, you have to include that in the title, in the file name as well. And also the metadata and file names should match as well, right? That you can go back and fix that afterwards, but that's basically it. So that's how that will actually speed up the upload process and clear some of the backlog. Hopefully clear some of the backlog from all of our scans. And I really want to just finish, if I have a bit of time, just to highlight some of the other bottlenecks that are probably coming down the pike, and it was brought up by Austin a little earlier, which is, I think we can put all this stuff up online, but until there is until we curate the data for the people who are going to be wanting to use it better, we're going to be limiting the amount of impact that these data have, right? And I know this well because this is what most of the overt data looks like, are these tomographs, right? And so people don't, educators and people who are just interested in museum specimens don't necessarily have the ability to turn these tomograms into a three-dimensional, object. They don't have. Well, they have the ability, they just don't have the training or the software access to do it, or even the knowledge to do it, right? And this has been quite apparent to me since the paper came out, and I've had so many emails saying, this is incredible, what a wonderful opportunity, this is going to change everything. I don't understand any of this data, how does it work? So it's been a little bit frustrating, to I mean, not frustrating, it's been really gratifying to see people use and excited about using this data, but understand that that, you know, just because we've made it available doesn't mean we've made it accessible, right? And I kind of think of it like, you know, having an amazing library, but if it's all in the language that nobody understands, then that's not accessible to you, right? You can't, it doesn't make any difference. And I worry that we're basically we've got the attention of the world with the publication come out now, and I don't want people to get frustrated by that. So I think one useful way of framing this and making this and ameliorating some of these problems is to think about these FAIR principles and applying these FAIR principles to our data. So findability, accessibility, interoperability, and reusability is something that we all think about all the time as museum people anyway, right? This is something that's baked into us because we're essentially just libraries with dead things instead of books. So this is something we think about a lot, but I do think we're not necessarily used to thinking about these kind of things with the breadth of use and audience that digital information will reach. So we need to think about that. And so because museum scientists tend to think about these things often, we find ourselves in a somewhat unusual position of being well ahead of the curve for other researchers and other research fields in terms of being prepared to do something about this. So the Non-Clinical Tomography User Research Network, or NOCTA, it's a hard-working acronym, is a research coordination network that was created by a number of people who are probably on this call, who actually, probably not surprising, are mostly museum folks, to build a community to apply these kind of FAIR principles to tomographic data, not just museum data, not just you know, our world, but across the entire breadth of basic and applied, basic and applied research, but also incorporating industry and educators and outreach. So if you're interested in this, is this going to work? Go forward? Maybe not. Oh, maybe that. Oh, there you go. If you're interested in this, the Nocturne Network group, if you feel like you want to have a say and have a voice at this table, a seat at the table, the Nocturne Network, or you can sign up for this here. We have a bunch of resources, including educational resources, an interactive map showing where all the available CT labs around the world are. We have an annual meeting every year. In person, this last one was at UT Austin, but this year will be in scenic Gainesville, Lake Alice. It won't be in Lake Alice, but I'm sure we'll find there at some point. And that's May 8th to the 10th. So if you're interested, do go on nocturnetwork.org and sign up. And thank you. I think I've run well over time, so sorry about that.
It's okay. Thank you. JJ's next. It should be under. It's, the issue is exact.
Food security, whatever.
Yeah, no, absolutely. No, and that's something, you know, it was just a bunch of like-minded people who put the front end, but the actual members of the network are very, and we're trying to get.
It would be wonderful to be able to put ourselves into human health,
Engineering.
pharmaceuticals, all those kind of from collection.
100%, yeah.
Okay, next is Jennifer J. Hill, JJ Hill from the Smithsonian National Museum of Natural History.
I can do that. Or you can use this.
Thank you very much. Hi, everybody. I'm going to be talking a little bit different direction today about the HIDA object replication project I did. Just want to point out that this is a photograph taken from Kimberly Nesbitt. She's a really cool photographer. Check her out. Or not. Yeah. Thank you. To appease the collections folks here, I've included a kind of a short snippet. please don't take away that this is like all the cool stuff we have. I'm not trying to show it that way. I'm trying to show you that I get to work for all these departments. I am the UTC specialist for CT at the museum, and these are beautiful collection photographs taken from the late photographer Chip Clark. He kind of got to go through all the different collections and pull them out. And at the time of creating this talk, these were the numbers. These numbers changed constantly. People are donating to the museum. They're also, the researchers are out collecting in the field for current, for wet collections as well as dry collections. So at the time when I created the talk, there were about 148 plus million specimens in the collections. There's 11 million item records online, and this is an initiative that the Smithsonian is trying to get more stuff available online for accessibility reasons. 292,000 of them are extant biological types. 139 are paleobiological types. Of those are mineral types, as well as there are about 1.5 million plus that have been geo-referenced. And anthropology is kind of selected because that's what I'll be speaking to you about today. So it's kind of hard to see, but it says on the top, considering non-biological specimens, cultural and ceremonial objects. So we had a request come in to the repatriation office from the hereditary chiefs and the Council of the Haida Gwaii Nation to help them replicate ceremonial objects in the museum's collections that they no longer have in person, but they want to kind of revitalize this cultural dance ceremonies, which were lost in the 1880s. So this request, I was given this request in October of 2021, and that was kind of at the height of the second bout of the pandemic. So the museum had a bunch of handling precautions as well as protocols for handling specimens. And I would mention that we didn't get to work on this, and so the initial request came in October, but I didn't actually get to see anything till November. I would also like to say that sensitivity and respect, and so next to the word respect is yakudang, and I might be saying that wrong, but that's the Haida word for respect. And when handling cultural objects and ceremonial objects, sometimes they can be really sensitive and really hard to talk about, but it's important to talk about and kind of get a consultation from the community. Here I'm presenting a case that's the intention of Case to Case to kind of use 3D technology to create replicates as well as that can be used in these reviving these dance ceremonies, as well as models for carvers and for educational purposes. You're also creating this archival record for cultural revitalization. So there's digital capture, there's physical milling to create the replicas, and the original objects were all hand carved and handmade on pieces of wood. And the idea is that we would be using milling to kind of machine make the replicas. And again, I said this to restore the ceremonies of the dance societies that were lost in the 1880s. And this also furthers the museum's of Natural History's IDEA Council to increase access to technology for Indigenous communities. And in this case, we use CT to explore the internal structure, the exterior surfaces. Specifically, I'll be talking about the paint to wood interface, as well as material composition, mechanics and functionality of the objects. And I would also just like to say that I have received permission from Guja, who's the hereditary chief of the Haida Gwaii Nation, from Sketdans, and the Repatriation Office Tribal Liaison, Dr. Eric Hollinger, to present the objects that I'll be speaking to you about today. So a little bit about the region. So it just says Haida Gwaii on the top. It's the former Queen Charlotte Islands. And it's kind of hard to see, but over here, Ketchikan, Alaska is just above the second blue island there. And the Haida Territory encompasses parts of southern Alaska, the archipelago of Haida Gwaii and its surrounding waters, but it's by no means a border. There are tons of tribes members and other people that are all around the world and internationally. This is where they're predominantly grouped. Two of the things that I'll be talking to you about today is a dance rattle from Masset, which is in the northern area. And it's this beautiful rattle that has two faces. It's a smiling face and kind of a sad face. And it's two halves secured with some leather string, as well as two beads and or stones on the inside to produce the sound. So when it's operating, it kind of makes a ta-dunk noise. One of the faces, I think with the smiley face, there's a kind of a condition of the rattle that was in. There's a broken part of the rattle. So there's a tooth that when we were scanning it, we had to kind of create them to be flush so that there wouldn't there's a crack in the wood, so it is kind of lifted up. And then the two dance whistles that I'll be talking about today are from Skit Date, which is more kind of closer here into the middle. Again, for both of these, we'll be looking at internal structure, possible wood grain detail to identify what the wood was that they used to carve the objects, as well as looking at for sound reproduction and function. So I told you there's a rattle, there's the beautiful rattle, and the two whistles on the bottom. These were all hand carved out of wood, and two of the objects were painted with natural pigments. And they were collected and purchased by Jane E. Swan in the fall of 1883. Haida art, if you can kind of see, has this beautiful, highly sophisticated form line system. So what I mean by that, Haida traditionally paint with three colors: black, which is the outline, so the outline of the face and the outline here, and it usually is made out of like soot and charcoal, and then red, which is the interior, which is like the nose and the mouth, and it's really hard to see, but on the whistle here, it's also on the mouth. It's to indicate like openings, and usually that red paint has mercury, lead, or iron. And then the blue-green color, which is the complementary color, you see this predominantly on the rattle, it's really beautiful, but it's actually can be really problematic when CT scanning. So it's possibly made of these light blue-green color because they're all natural pigments, possibly may contain light reactive minerals such as selenium and vivianite, and when they're exposed to ionizing radiation, actually can change visibly in color. So this beautiful light blue color may darken to a dark blue color. I bring this up because it's not reversible. You can't just kind of scan the object and then it would appear, it's not temporary, it's permanently damaged, which can be a problem. And then also to bring it up that you wouldn't want to do that from a museum perspective and from a collections perspective, because you change the integrity of that object and you're no longer making a replica of the exact object, which is what they asked for. You're actually you've damaged an object and not creating it according to their standards. Thanks. So I said it was problematic. So we had to do a couple things of analyses before we could actually scan the objects. So my colleague, Dr. Hollinger, who's also the tribal liaison I mentioned, he used portable X-ray fluorescence on a little bit the dance rattle specifically, because it was the only one of the objects that he did scan that had this blue-green color. And so he spot tested the nose and he even spot tested the mouth where this beautiful red color is. And the results of the spectral analysis basically show that it has mercury, which we kind of knew when you look at Haida objects, even Tlingit objects, anything that with red paint usually has mercury in it or high iron. So that was expected. But then when Dr. Hollinger did a spot test of the rattle with the blue paint, we noticed that it had high iron and high potassium, which was interesting. But we also rolled out from this analysis that the blue-green color wasn't vivinite, but it was probably seladanite. And how do we know that? Because the spectra showed this high iron and potassium, but it wasn't phosphorus. So vivianite, when you look at it, if it's phosphorus, it glows and it didn't have any of those properties. But it's still possible that seladanite was present. But I will mention here and then from the next image that we didn't see high magnesium. So when you do these kind of tests, you would expect high iron and potassium, but we didn't see magnesium in the patch test that we performed. So there was one more analysis we had to do. We mimicked high mineral compositions of naturally pigmented paints. So we used two substrates, pure chloride and copper chloride, and thank you, and a silver metallic premix paint to represent synthetic paint, where the other two mimicked the natural pigmented paints. And we used, each substrate was painted on a piece of wood, and there's nothing special about that. It's a branch that my boss cut in the workshop.
And then I applied substrates in 1, 2, and 3 coats using water, as well as 1, 2, and 3 coats using glue. Most naturally pigmented paints are binded with some kind of binder, whether it be resin, paint, glue. So we were trying to mimic that. So mercuric chloride and copper chloride do not play well. So there was every intention not to overlap these things because they should not touch each other. So the mercuric chloride was the iron ochre analog for the red paint. And basically it soaked into the test wood. There wasn't any color reaction, which was good because the rattle has red on it. And we also want to make sure that didn't change color. But we did notice there were small grainy crystals that formed at that wood surface interface where it was painted on and into the wood, but we didn't see anything that would alarm. So we used copper chloride to mimic the possible seladanite in the blue-green paint. And it was different than mercuric chloride. It adhered to the surface, but there was minimal absorption by the wood, which is a good thing. And we noticed no color change, which was really great for us because that means we could probably scan the rest of the objects. And then this silver metallic paint, which was supposed to represent synthetic paint. It wasn't good in either circumstance. It was flaky when it dried off, and it didn't actually adhere to the wood very well. And it's this very dense kind of material that you can see in the X-ray projections. But the good thing of doing this test and making sure is that we saw no visual change so that we can do it. So I'm faster, sorry. Thanks. Okay, so a little bit about the object packaging and transport. I might breeze through this, is that a custom kit was created. These objects are located in the collections at the Museum Support Center in Suitland. And basically the collections people created a lovely custom crate. And they wrapped all of them in foam and string, and you can see there's tissue. This is kind of an example of the rattle and one of the whistles unwrapped, as well as what it looked like when we mounted them into the CT. And I'll just say a colleague helped me move those from the museum. These were the original segmentations that I created of each of the objects, of the whistles and the rattle. And these were provided to the Smithsonian exhibits team. And I included a little bit, I told you in the beginning, we didn't know if there were beads or rocks, but it turns out there are 2 rocks inside of the rattle. And they look to be from the shores of the Haida Gwaii, so that's interesting. Thank you. These are these beautiful conceptual models that 2 of my colleagues, they're both 3D artists, Caroline Tome and Chris Halchwander. They work in exhibits. They digitally created the objects and they digitally split them so that we could see the interior of the objects so that they could be moved. Yes. So where are we in this project? Well, it's actually still going on. Guja, the hereditary chief, is working with the Department of Forestry, the University of British Columbia in Canada, to mill these objects. And milling began in 2022. And the top portion here is cross sections. It's a little hard to see cross sections of the wood grain. So Guja's sons are master carvers, and they can actually tell by looking at cross-sectional views of wood what exactly the type of species that was used. And so they're still doing that. But I wanted to include it because it's very interesting because I was able to see that when I was making the segmentations. And then this is a robotic arm that's actually milling an object here. And then that's kind of a result of one of the objects, not one that I'm specifically talking about, but that's what they look like. And then I just like to acknowledge my colleagues. I only had this for a small portion and it's still being done, but specifically Guja, thank you very much for letting me present this, as well as Dr. Eric Hollinger, the tribal liaison. And I'd like to thank the artists, Charlie, who was the contractor, who helped me move the objects from Suitland to downtown, and my boss. Thanks.
Yeah.
Yeah.
Okay. Next is Mateo Capri from the Field Museum.
Okay. Okay. Thank you everyone for having me here. It's my privilege to present you some of my research that is going on and will start in summer in my lab at Johns Hopkins. Obviously, I don't have to explain to you why phenotype, what it is, and why we like it. But maybe I can remind you why structure and morphology is so important. And the reason is that structure is the only thing that connects the history of life, extinct and modern. Molecular work can go just a few million of years, therefore genomes and molecular data are only restricted to more recent species. Therefore, morphology is the only way we can bridge the history of life, where the fossil record is the representation of the majority of diversity that ever lived on our planet, and therefore it's fundamental to understand the origin of clades and to date the origin of clades, and also to figure out where these clades actually appear. But on the other hand, the fossil record is incredibly difficult to interpret unless we understand what's the connection between morphology, ecology, behavior, and so on and so forth using modern species. So in other words, the fossil record and modern diversity are complementary faces of the same coin that depict the history of life. And we can basically put together these sources of data through quantitative comparative methods and 3D imaging. This is because not only we can extrapolate information from the three-dimensionality of the structure, but we can characterize this and put together these sources of data because the structure has an evolutionary history that makes structure homologous. But we have a problem regarding the modern species because until now, major efforts have been done on adult phenotypes, trying to capture the evolutionary diversity of life. But there is an incredibly important resource of data from extant species that is still way behind, and I would say the most behind source of data in terms of collection, and this is developmental biology. In other words, embryos have been fundamental to understand how morphology arises generationally, but it also follows specific rules that have been the scaffold for the history of life since its beginnings. And the general reason for that is because we can actually track morphological change through development, starting from the phylotypic stage, which is this moment in organogenesis where morphology is extremely conserved, and only later on in development, the clade and taxon-specific traits that characterize modern diversity is actually arising. So this means that if we put together an evolutionary framework that includes the early moments of development, we can actually bridge evolutionary biology with developmental biology. And from a molecular perspective, we are doing spectacular because we had the Vertebrate Genome Project, one genome sequenced for every single vertebrate alive today. Spatial transcriptomics is bridging gene expression with morphology. And CRISPR-Cas is the experimental attitude of twitching the genome to understand how a different genome can bring different phenotypes. But when we think about phenotype per se, we are far behind, and this is mainly the kind of leftover of the tradition between the 50s and the 90s, where Alchan blue and alizarin red is the main way to quantify and show skeletogenesis. The problem of this technique is that all the soft tissues are completely erased, and the skeleton per se is only one part of development of the organism per se. But there are different ways, as you all know, that we can use. So 8 years ago, I decided to go through the museum collections and looking for non-model organisms that can actually let developmental biology move over from the 5 model organisms we use since Aristoteles. And the museum collections are the only place where non-model organisms are actually created, because due to the collection of endangered species and species that are actually today extinct, in your collections, you have embryos of animals that are completely impossible to sample today. And therefore, I decided to go in the collections of fluids for bats, for primates, as in this case. And it's insane to see how samples like these Hylobates, still completely numerous, has here a full embryo in complete development. And this kind of samples is not only telling us about developmental biology, it's telling us, for example, how the evolution of the interaction between adult and embryo evolved in primates, including our species. We can also go towards a synchrotron, and I hope this works. So the synchrotron for smaller samples, this doesn't require staining, but allow us to go to subcellular resolution in terms of embryonic samples. And in case we are lucky enough to get fresh samples, I know that some of your people in the museums are going around for fieldwork to get new samples. Sometimes the females are pregnant. In case of this kind of fresh sample, we can use a technique that was invented in 2013 in order to image the structure of the brain. And in this case, this technique takes out all the lipids from the sample, and then we can use primary antibody and fluorescent antibodies in order to image in 3D the early developing structures of the skeleton. And this is one of the examples where we have a single embryo, but I can put multiple antibodies targeting different systems and image this in 3D. And this can be done across one developmental series or multiple developmental series in order to track what are the small changes early in development that are responsible for the extreme phenotype of the adult. And this is an example, very qualitative, but here we have a shark, and here we have a skate. And this is one of the early moments in development where we are starting to see this divergence in the phenotype. And this is how it looks later on in development. So our skate that has here the pectoral fin, like any other chondrichthyans, grows towards the extreme phenotype. Here, I just want to show you how you can image this at the cellular level. So we are talking about centimeters long samples that are cellular level accuracy. And so if we do this, including CT scan, synchrotron, and clarity and immunofluorescence across the tree of life, at this point, we can actually infer how development influence evolution from a mechanistic level, which is something that is not possible just looking at modern diversity and the fossil record, just because we have to understand what are the constraints in development that dictate the rules of phenotype. And at that point, we can infer why different phenotypes can indeed appear. And so in the last 8 years, I collected almost 900 embryonic samples that belong to 43 different species, spanning from chondrichthyans to humans. But obviously, this is just a proof of concept. And so what I'm trying to do today is to open a call to all of you, saying that if you go in your fluid collection and you find embryonic samples, I would be more than glad to collaborate and figure out how we can expand our sampling within the tree of life. And as a plus, all these scans, independently from the method that you are using, are 3-dimensional. So what this data mean translated is that we can actually investigate the history of life in 5 dimensions. through our time, development and evolution, and free our space, the morphology per se. And so with this, I want to thank all my collaborators and fund resources, and thank you for your attention.
Okay, John Woodward is next. One more talk before our last opportunity for Q&A. Awesome, thank you.
How we doing?
Thanks, Ed.
Pension flagging, hanging in? I'll be fast.
Advance the slide and that's the list.
Okay, thank you. I'm John Woodward, digitization manager at the Museum of Comparative Zoology at Harvard. And I'm going to be talking about 3D reconstruction of embryos from histological serial section images. And I'm mostly talking about this cute little bat here. This is Special Collections number 1995 in our collection. So for those unfamiliar with the serial sectioning process, a specimen is embedded in paraffin or some other kind of matrix and then sliced into very thin sections with a microtome, sort of a glorified ham slicer. And then then the sections are laid out in sequence on the slides and stained with something that will add contrast to the anatomical features that you want to look at, and then covered with a cover slip. The reconstruction process that I'm going to be talking about is in some ways an incomplete reversal of sectioning. High resolution images are created and then aligned and stacked. The reconstruction process opens many avenues of analysis, including segmentation and measurements, creation of 3D models, all kinds of other stuff. I'm guessing that the machine learning folks are going to have lots of thoughts about what can be done to improve the process here, but also what can be done with the results. History of reconstruction, heavily abridged. On the left, you can see a model. This is a model created in physical space using a system of stacking wax plates. But it's basically the same process. The images of the sections were projected onto wax, the cutouts were made, and then you stacked the cutouts. On the right, I've put a 3D surface model made from a micro CT scan. Micro CT reconstructions result in a stack of registered slices of density information. In some ways, this is a born digital version of the stack to plate process. So recent research. Research scientists are still actively using and refining this technique to understand aspects of anatomy, evolutionary development, many other disciplines. I wanted to draw particular attention to a really fascinating paper by Benoit and Jasnowski, where they did drawings of the grinding face of a fossil and serially ground it, did a drawing at every stage of grinding. Realized partway through the grinding that it was a holotype of a new specimen that they were working on. So they continued with drawings, did very detailed images of the drawings, and then reconstructed both virtually and with a 3D printed model. So value for natural history collections. 3D reconstruction represents a new way to make the specimen discoverable outside the collection. It can also yield information about the specimen itself. For example, the horizontal striations visible in this cross section here give us a clue about staining technique, which we hadn't noticed before. The number of horizontal bands corresponds to the number of slides in this series. And we think that the darker areas of those bands are indicative of more stain being applied to the first sections on the slide than the final sections. So you end up with this kind of like real striated appearance. My work has focused primarily on the Harvard Embryological Collection, also known as the Minot Collection, for its primary preparator, Charles S. Minot, and his students. It was created over several decades in the late 19th and early 20th centuries. The preparations are excellent and the metadata is really, really complete, which turns out to be really important. And most of the series have a really nice, either a line drawing or a photograph, depending on the time when the preparation was done. And in either case, it's very important, having that photo documentation, or that the prior knowledge, basically, of what the embryo looked like before it was sectioned up. So many collections hold serial sections preserved on slides, but not every collection has access to thousands of dollars worth of software and computing power. Therefore, I felt it was important to make this process work on my laptop using free software. So I used ImageJ and 3D Slicer as my primary software packages. Once acquired, the images must be, image acquisition, the first part of the process. Though the scans were done at very high resolution, this need not be part of the reconstruction process. The scan images were significantly downsampled, then individual slice images were segmented out manually. All credit goes to my colleague, Emily Blank, for this patient work. I think if going into the future, I would want to look into some machine learning alternatives for doing it, because it's very time consuming. Once acquired, the images must be registered. In most algorithms, this is done by matching pairs of images, beginning with a reference image and moving both directions through the stack, as seen in the top diagram. So you choose a fixed reference image, then move the adjacent one to match it, and then proceed pairwise out from the middle. The graphic on the bottom progresses rapidly through a sequence of sections from the bat embryo. You can clearly see the difference from before and after. Image registration is done by applying one or more transforms to the moving image in every pair to make it match the fixed image. The most common types of transforms are shown in the table here. I'm mainly going to be talking about the widget, which is translating in XY space and also rotating, and the elastic, the last one listed here, where there's sort of mesh deformation that you can apply to make the pairs match. When reconstructing specimens with any amount of curve to them, the original shape is unattainable without prior knowledge of the specimen. This is referred to in Mendelein et al. As, quote, the banana problem. As shown in their diagram, when the banana is sectioned, if the section images are unaffixed to a reference point, the pairwise matching of the faces will result in a cylinder. And on the gray banana there, the spectral remnant on the right is the banana reconstruction that could have been if only we had prior knowledge to compare the section images to. But there are further inherent difficulties. Process of cutting and preserving sections results in warping, tearing, inconsistent staining, changes over time, and other, quote, artifacts which alter unpredictably the topology and extension of anatomical structures. Because of these deformations, it's unlikely that any given section can serve as a reliable reference for the registration of the others, and it's impossible to know anyway, absent prior knowledge. In other words, registration of serial section images is inherently difficult. Elastic registration is likely to compound the lossiness of the sectioning process, and perfect registration may be impossible. All that said, many smart people have worked the problem from different angles and have written software based on resulting algorithms. I recommend the linear stack alignment with SIFT as one plugin that's easy to use, gives good results, and works quickly. It's limited to linear transformations only. It doesn't do elastic. Second, TrackEM2 has a baffling array of menus and options, but it's worth it. It can register with linear and elastic transforms as separate steps. So you can do one linear to sort of line things up, and then a separate elastic transform to like fine tune your results. It has no RAM restrictions, so it's very good for limited computing power, and it doesn't alter the original data. I prepared a sort of stepwise click here, click there kind of protocol for using TrackEM2, and that link at the bottom will take you to that. So what can go right? Or can you start the movie up there. Yeah, thank you. So this is the result of rigid registration of 732 bat embryo slice images. The original line drawings and the slide label metadata, the original line drawings, these guys on the right. And especially the slide label metadata provided a helpful reference for the slice interval. That is how far apart the slice images should be placed. And thus how pudgy or stretched out the bat embryo should be. And then from here, the stack can be cross-sectioned in any arbitrary plane. It can be measured internally and externally, cleaned up and smoothed for 3D printing, all of that stuff. So in natural history collections, we use the language of science, but fundamentally we're in the memory business. And I'm thinking about specifically Dave's question or this sort of formulation from before, like who is it for? I think that it would not have been possible for the folks who prepared these slices to know that, and especially who made very detailed drawings to know that these would be important for reconstructing in a sort of virtual space. But we can't know what our work is ultimately for. It's tempting to think of this kind of thing as like a techno apotheosis of the specimen, but I see it more as like a bridge between islands of memory to connect both from the sections to between the sections and the line drawings, but also to unknown uses at some unknown point in the future. And that's it. And I'd like to thank those folks who helped with various aspects of this project. That's it. Thanks.
That was a lot of presentations. Thank you so much. And thank you for everyone who's still online. We're gonna take about 10 minutes. We have we have about 10 minutes for question and answer. So if you have a question, please put it into the Q&A. And I'll turn this around. That was
are they consistently thick, the slices? Are they always the same? Or are you assuming that they're the same thickness when you will redesign?
In the case of the one I did here, yes, they're always the thickness. The slice thickness is typical of this preparator. Every slice has the same slice thickness, and usually the number, like range of numbers of slices on it. So it's really meticulously. Other slides that I've looked at are you get worse results.
Okay, thank you. Question two for John. Did you try throwing those image sets through a photogrammetry software? Because a lot of like reality capture uses SIFT to do alignments, and was wondering if, I don't know, have you tried any of that? I was just curious what some of the results might be.
I would be too. I didn't try any photogrammetry software. I tried some really initial exploratory data sets with Mira, which has some alignment capabilities and whatever, and kind of ran into the sort of same similar problems to using ImageJ. It's a lot of trial and error, and you get different results depending on which reference image you choose. And yeah,
I'll try it for you.
Okay.
We can just plug it in.
Yeah. Okay.
Yeah, that'd be cool.
You're noting for your staining, did you know the differences staining for 5% versus 4%?
Sorry, can you say that a little louder so they can hear you over?
I was asking if the preparation type, the fixation preparation type for wet specimens, if it's 95% versus like strictly versus the combination of formalin with an ethanol type, impacted how well the iodine is staining.
Yeah, so unfortunately, I don't even have access to that information, so this preservation history most of the time is unknown. Like right now, I know they're all at 70% ethanol, I know that much, but there's no record of like whether it was formalin fixed or ethanol fixed. And that's something that like with the DICT, a future investigation that I would like to do would be to like purposely try different fixing methods, but also purposely like treat things the way that you shouldn't do it. So there are a few things like multiple freeze-thaw cycles can really affect tissue quality. Having a gap between when the thing died and when it was fixed, those kinds of things where like I can look at a scan and say it's poor quality and it might be because of this, but I don't know because I don't know what caused it. So that is a future direction that I would love to go.
Are there any online questions? I had a question for JJ. So you might have mentioned this in the beginning, but it seemed like there was a lot of issues with doing the CT scans for the reconstruction, at least with like the pigment and the issues of, you know, some degradation of color and stuff like that. So going back, would you try maybe just utilizing photogrammetry and light scanner? Like, was the internal information really?
So the objects were originally, or I think they were actually they did photogrammetry and they did imagery, traditional imagery, but the problem is, I didn't get to talk a lot about it, is the functionality. So these are dance whistles that they're not made by blowing, like they're made by blowing and they're also being used during a ceremony that they're dancing and moving around with. So for functionality, they really, for the milling purposes, needed to see the interior of the objects, but they couldn't take them apart and so that's why we ended up using CT. The problem we had was paint was the problem because with biological specimens, most of the time, you know, you're not having to worry about if there's like impacts on the scales because you're not looking, unless you're looking specifically at that, you're not looking at that. So for the cultural items and the ceremonial objects, paint was a prominent factor. So there's also like research on papyrus scrolls as well as in Egyptian research with paints on the outside of sarcophaguses. You know, you all have to think about that. It was just an extra step that we did to just make sure we weren't going to alter the objects. The scanning itself relatively was easy. And then we did that substrate analysis to just reconfirm and also to do test parameters. So we did it at like 90 kV and we do it at like 130 kV upwards up just to test the parameters to see which one would be best used. So I think they were traditionally photogrammetry and images were taken. They wanted to know the functionality. We would have never known they were river stones. They thought they were beads because that was traditionally what was used inside the rattles.
I really enjoyed those talks, just brought together some research applications I'd never thought of before. So I have a question for Mateo. The 900 embryos you found, were those all completely research specimens of the museum?
Not all of them, but the majority of them. Okay. So regarding alligators, turtles, chicken, model of more reasonable organisms, I managed to get the eggs, and then I fixed them in the lab, and it's exactly the problem of having fresh samples. Regarding primates, cetaceans, so on.
So I assume we get the embryo data, the specimen was scanned. They weren't scanned for you to do your work, they already scanned.
No. I went there
so you
to scan them for my work. Yes. And the idea is that you can still do it in museums.
Right.
The main point is, although my interest in selecting samples was driven by my research, I know that this could be a project, a collaborative project for the next 5 years that could really bridge the two things.
Right.
Because obviously I'm just one, so I'm prioritizing what I need. Right. This could become a resource for everyone because there are very few labs doing this kind of work. And one of the main reasons is that having animals in the lab is extremely expensive and the sample between the three is extremely low.
I guess I was thinking just for some tax, it may be easier to tell the specimen during an embryo. During it far along in pregnancy. For others, not, I mean, discovery, internal embryos, surprise, right? So I'm just wondering if there's some like pre-screen.
The best way to pre-screen is to see if specimens were dissected before. There are many researchers who just went to think how we can read their lungs and stuff like that, and never paid attention to what was inside the specimen. So like a few years ago, I asked to scan this adult sphenoid that was regrowing the tail, thinking that the tail was cool and urine, and then it comes out that the sphenoid is completely ovulated. So it has dozens of applications similar to the yes, so you can never know, but I think it's worth it.
We have a question online, and it is, where can you find information on the setup/operation of Sugar?
Yeah, Sugar has a GitHub page, just Google Sugar and GitHub. There is pretty decent documentation on it, it's a little tricky. There is also a thing called SDF Studio, which also has a GitHub that I might actually recommend over Sugar, that we found later in our process, that has a lot more of that interoperability kind of built into it, a bunch of algorithms. So you can test like news against Sugar pretty quickly. So yeah, SDF Studio on GitHub.
Yeah, and in case you didn't hear that, I will say it louder, closer to the microphone. There's a Sugar GitHub, and you can also look at SDFS, which is something that was found a little bit later in their process than Sugar.
Yeah, okay, we're gonna take a hour break for lunch. We'll leave we'll leave the Zoom open, and then we'll be back at 1:30 this afternoon with Nick Pienson. So thank you for everyone who joined this morning, and thank you to our speakers. Start the recording. All right, everyone, welcome back. Up first today, we have, this afternoon, we have Nick Pienson from the Smithsonian National Museum of Natural History. So I'm going to turn it over to Nick, and as a reminder, if you have any questions along the way, please feel free to put it into the Q&A section at any time, or you can raise your hand or ask the question once we get to after the series of speakers. So thank you so much. The recording is started again, and take it away, Nick. Thanks.
Sure. Can everybody hear me? Thumbs up.
Yes, sounds great.
Okay,
Thanks.
great. Okay, good. I'm really happy. I wish I could be there in person, but I'm a new department chair, and those responsibilities are really kind of consuming me right now. So I'm coming to you virtual, and what I want to share in 10 minutes is 3 quick stories, and there's a thread through all of them, and the thread is both familiar and unfamiliar, maybe. The familiar part being that it involves digitization, 3D digitization, but it specifically focuses on fossils in the field, vertebrate fossils that are too big, too fragile, other considerations that prevent us from removing the objects and bring them back to a museum. And so the challenges of what you do in those situations there's some common challenges there. And I now have 3 case studies. I'm working on my 3rd one right now, and I want to share them with you in 10 minutes. And so I'm going to keep my eye on the time here, and hopefully draw a bigger picture, a bigger takeaway about the utility of these kinds of sites for sharing with other audiences. My specific audience that I want to share with are other stakeholders and decision makers in other countries who may have a lot of influence on science collaboration, capacity building. So the first thing I want to say is that science is done by people. It's not done by machines. And your capacity to do science is directly related to the kinds of networks that you have and the collaborative teams that you can build. So the work that I'm going to present involves collaborations with many people. Some of them are mentioned here. Some of them may be familiar to the audience. Holly Little, for example. John Blundell, also from Smithsonian, but from a different program office. Huge appreciation to the Digitization Program Office, who provides so much expertise and resources for getting a lot of this work done. The big scientific frame for a lot of this work has to do with the repeated invasion of marine tetrapods into ocean ecosystems over the last 250 million years from many different lineages of tetrapods, reptiles, mammals. They've gone back to the water from different ancestries in different times and different places, and in many cases have achieved common solutions. And those solutions are both functional at the tissue level, even down at the subcellular level, but also up to the scale of ecosystems. They've convergently achieved top positions in food webs. And so it shouldn't be a surprise that we might find similar patterns in the fossil record that reflect these convergences from cell all the way up to ecosystems. And the first one that I want to share that really I'm going to go through chronologically in my own chronology, not through geologic time, but this is now almost 15 years ago at a site in the Atacama of Chile called Cerro Baena. And the deal was there was a road expansion along the Pan-American Highway in a remote part of the Chilean desert in the north. And whale skeleton after whale skeleton was uncovered by a road construction company. There wasn't much time to document all the fossils in their arrangement, so we 3D digitized them using a variety of tools that, for me at least, in my own experience, were experimental. It was a new thing for me. And we were able to capture a lot of information in a very short period of time. And that really changed the way how I thought about the utility of these approaches for definitely documenting very large vertebrate fossils, but also then for sharing them. So if you do have your camera, point it at the QR code, not a joke. It should take you to exactly this webpage, which is part of the Digitization Program Office's 3D Voyager interactive platform online. And what we were doing here is sharing a snapshot of research in action, but it shows basically an entire whale skeleton, nose to tail. This is late Miocene in age, 7 to 9 million years ago. This is one of the 40-odd skeletons. And so it documents the articulation, the taphonomy, it's all right there in a way that we can't access now, actually, because this is still in a plaster jacket among many other dozens in a museum in northern Chile. And so this provides a conduit for research activity that is kind of like an extended specimen. This does have a specimen number, so it does qualify in that kind of rubric, but it was in situ. And so the fossil is no longer in that place, but we have all this digital information, and the durability of it here almost 15 years later is remarkable to me. And that says that we kind of did it right using a variety of tools, but I've been interested in applying these tools to other places in other times. And that led me to ichthyosaurs. I've worked with several colleagues who've kind of opened my eyes to the entire world of the Mesozoic and what a parallel it can be to what I know from the Cenozoic, from the marine mammals in the Cenozoic. There were a lot of lineages in the Mesozoic that were doing the marine mammal thing then that marine mammals are doing now here in the Anthropocene. And what's interesting is some of the patterns are the same, some are different. Ichthyosaurs, for example, seem to have achieved very large body sizes very rapidly. And at the time that Lena Delstead and I wrote this commentary for Science, it seemed like the major pattern was that whales take a long time to get very large. But then last year, this giant whale from the Eocene of Peru was discovered, Parasitus, which seems now to be about sperm whale sized. Whales achieved that gigantic body size in about 10 million years. It's still ichthyosaurs beat them by a magnitude back in the early Triassic. So there's, again, looking at these patterns of convergences, it's not exactly the same, even for groups that we largely think of having achieved broad similarities. And this is where the ecosystem level convergence is really interesting. There's a site in Nevada that preserves several large ichthyosaur skeletons from the late Triassic. These are ichthyosaurs that belong to the genus Shonisaurus. And this is specifically at Berlin Ichthyosaur State Park. So it's a shelter over a quarry that was exposed almost half a century ago, a little more than half a century ago. And many of the beautiful bones that have now been oxidized to the air. So it's actually really hard to tell bone from rock. And it was still spread out over such an area that we thought this was an appropriate use case for 3D digitization. You can see John Blundell on the right side of this figure of this pano, Holly Little's off to the left. And the result on the next slide, this is from the eventual figure on the paper describing what we think is going on at Berlin Ichthyosaur, which is a lot of whale-sized ichthyosaurs are all preserved closely together. And the modalities that we see of body sizes are all extremely large animals, and then some neonatal fragments that were found interspersed. There's actually several bone-bearing horizons at the site throughout the canyon in the rock formation of the West Union Canyon in the Luning Formation. And we don't think it's related to any kind of environmental catastrophe, but instead it represents a pattern that we've seen in marine tetrapods going on for at least here, we think, setting the antiquity at 230 million years of feeding in one area and giving birth in another. So we basically thought that Berlin Ichthyosaur were making the argument that it was a nursery of some kind. But to share that 3D information, because again, it's really hard to tell rock from bone, we wanted to use the best data set we had, which was an orthographic or 3D reconstruction of one of the main quarries. And we had to label it up. And this took a lot of time to do right, because there were a lot of bones. Marine reptiles have so many more bones than marine mammals. And I think it was worth it. So what we created was a scrolling website. Apologies, I went through the last slide too long, but just enter or just type in enter the sea dragon, and you should find that 3D website. And the scrolling website that goes with it, that goes through the 3D model. So again, you know, we think these data are reflective of a pattern in marine ecology that's been going on for 230 million years of separation of reproduction and feeding. And the scrolling website was kind of the next step of sharing that 3D information in a way that told the story. You know, it's all about narrative and trying to communicate that. And the third part, I'm realizing I'm coming up short on time, has to do with sea cows. So sea cows, like many other marine reptile or marine mammals, once had terrestrial ancestors. And so this is a four-legged sea cow that's on display here at the National Museum of Natural History in Washington, D.C. You can go into the Deep Time Fossil Hall and see the skeleton of Pisosiren. It's a sea cow, and it has 4 weight-bearing limbs. But one of the really interesting things about sea cow evolution is that they seem to have diversified through time. And one of the typical patterns we always see is we always find many species of sea cows wherever we look for them, except in the modern world. So it's a little bit of a weird case where the modern world is tremendously different from the majority of the evolutionary history of this group. And in Qatar, in particular, there's a really interesting book with the modern species that's still around. One species, dugong, in Oceania, and one of the second largest populations of dugong is in the Arabian Gulf, and nobody knows anything about it. This is a screen grab from a drone taken a few years ago, showing the density of dugong that live in the Arabian Gulf. Very little is known about their life history, and it was through some collaborations with colleagues that I had who were in country in Qatar, where they said that, you know what, there's also some fossil sea cows in Qatar. Why don't you go look and see? And the reason why it would be really interesting is we want to know if there were multiple species of sea cows, because that's something we always find in the fossil record for this group. And while we haven't really found all of these multiple species, one thing that is pretty amazing that we have found is a very dense site of sea cows that I think exceeds the density of bones that we've found in Sarabaina in Chile. So it makes it one of the, I think, the densest marine mammal fossil formations, rock units, but certainly one of the densest fossil sites that we have yet documented. It exceeds Wadi Hitan by several magnitudes. And this is the application of 3D tools, because we're not able to collect every single one of these skeletons. There's, we documented over 140 localities in about 1 square kilometer. But what's great is knowing that I can use photogrammetry tools, I can create these 3D data sets really rapidly. So the same kinds of approaches we were using in Chile, we now can do this even more quickly, and using someone, me, who doesn't really know exactly what they're doing with a camera rig. I can learn fast enough and I can pull out a data set that's pretty good. These are orthographic images. So it is a 3D model, but you're just looking top down, and there's no parallax here. And the detail is really striking. So this is important because a lot of these skeletons cannot be collected. They have to remain in place. And we're able to document so much, not just about their current condition and preservation, but also we can know more about their taphonomy and know how they came to be. So obviously this kind of in situ using digitization in the field allows you to split up, parcel out the kinds of work that you do, capturing information in one place and then taking it back to the lab. But of course, there's a lot of questions about how we preserve, with the information that we preserve here, how we archive it, how we make it conform to data standards that are widely available, and of course, sharing them. That access and benefits sharing is really important. And in Qatar, we were able to elevate that because there's such an emerging interest in data sharing. So last year, a lot of our collaborations kind of snowballed into a larger informal network, and that's why we call this a Data Diplomacy Unconference. But we're actually able to host this along with our Qatari colleagues at Qatar University. And so happy to say that this involves scientists from every Gulf nation. And this is a part of the world where obviously not everybody always gets along, but it's in those moments of tension and that you can have surprising diplomatic connections emerge. So fossils for diplomacy actually can work in some places and in some situations. And why that matters, because some countries like Qatar actually have a large role to play in keeping the carbon that we need in the ground to stay in the ground if we want to meet our 2030, 2040, 2050 targets. So by trying to create better bridges for data diplomacy and data sharing, I hope that we can have a role to play in this kind of, in a positive outcome. So that is it. I realize I'm out of time, I think. Is that right, Jill? Looking for you to nod. Okay.
Yeah, thank you.
Yep. Thank you all. Wish I was there.
You have any questions?
I can stop sharing if you want, Jill.
Oh, yes, that would be great. Thank you.
They might have wished it was muted after this. But hi, I'm Kit. I am, well, most of you probably know me from iDigBio, but I'm also a PhD student at the University of Colorado. where I work in information science and interdisciplinary quantitative biology. And in interdisciplinary quantitative biology, I'm helping out on this NASA Bioscape project, where we're using hyperspectral and multi-scale imagery to understand biodiversity and invasive alien species in South Africa. Unfortunately, these are not extraterrestrials, even though we are working with NASA, it's trees. So, yeah. Can you click on it again? I think it might have. Perfect. Okay, so Bioscape stands for Biodiversity Survey of the Cape, and this is actually NASA's first biodiversity field campaign, where we're incorporating airborne imaging, spectroscopy, LiDAR, and field observations. And this is on the greater Cape Floristic region, which we are including the coastal and marine environments in the scope of this grant, but I'm just going to be talking about a very, very small portion of this grant. So what's interesting is this area is one of the world's 35 biodiversity hotspots, and 69% of the estimated 9,000 species that comprise the GCFR are expected to be endemic, and parts of it are UNESCO World Heritage Sites. And it does comprise of about 4% of South African land. And what's super crazy and why it's really important to protect this area is 67% of the country's threatened plant taxa exist in this small area. So, and that is the fynbos vegetation, which is an Old Dutch word for fine bush, and it's very well adapted to grow in this nutrient deficient soil and very tough climatic conditions. Unfortunately, there are 4 main invasive species that are starting to overtake the Cape, including pine, acacia, eucalyptus, and hicea. And where this is a big issue is especially, like lots of issues, but the fire regime is particularly impacted by this. One, pine fires are very, very large. I think I put it here, yeah. So typically fire seasons with fynbos are about 3 to 5 tons per hectare of biomass fuel loads for burning. And then pine fires take up to 10 to 25 tons per hectare. So not only are you seeing these very detrimental fires, pines also consume more water. So you're taking more out of the water table just by them existing, and then causing more catastrophic fire events. And we're kind of hoping to see how we can you click on it? Yes, thank you. And we're using imaging platforms. So how we're getting this imagery, we are using 3 scales. The one that we're focusing on the most is this middle scale, which is AVIRIS data, PRISM data, HEITS data, and we're using the NASA G3 and G5 research aircraft with 3 imaging spectrometers. So we're using, these first 3 are spectrometers. These are both in the EMR, so the electromagnetic range. This is an emissivity metric, so looking at thermal emissions. And then we're also, for structural, using the Elvis LiDAR system from NASA. And how this kind of all incorporates into imaging is we have people on the ground. We are potentially putting drones in here, but we haven't done that yet. So right now we have people on the ground, below cloud aircrafts, and then satellite imagery that's all coming together to kind of understand invasion at a larger scale. So we start at the ground, and we are taking samples, validation data, finding out where the invasive species are. So when we do go up to this AVIRIS data, so we're using like support vector machine learning classifications, random forests, like a bunch of different machine learning algorithms to kind of classify where these guys are, and then apply that to other areas of the ecosystem that we don't have imaging on. And putting it into this middle layer, which is where we're looking at spectral and structural diversity. I'm mainly looking at spectral diversity, and we're using an algorithm based off of a paper published in 2020 called the La Liberté algorithm. It's an amazing paper. I would honestly recommend reading it. But what we're doing is we have these hyperspectral bands, so about 5 nanometers wide each. We have about 200 bands. In this system, if you're familiar with NEON, their hyperspectral instrument has 426 bands. And what we're doing is reducing it down to 2 or 3 bands via principal component analysis to kind of get spectral diversity metrics for these areas. And then pulling that out to look at how diverse an area is. And this next slide will kind of clarify that. So based on the algorithm, if you can pretend that this is a pixel from MODIS, so MODIS takes images at 250 by 250 meters, and we are putting it into communities of 50 by 50 meters. And in these 50 by 50 meter communities, we're overlaying the 5x5 meter AVERS data that we have to get the spectral diversity in this area. What we're trying to look at is overall diversity, which is gamma diversity. Alpha diversity is what is the diversity within one single square, and beta diversity is in between communities. And where this is kind of important and super interesting, in my opinion, is there is a hypothesis based off of this grant, and what we're looking at is the more spectral alpha diversity you have and gamma diversity, the less likely there is an invasion happening, because that species hasn't been able to come in and overtake the area. So looking at really like broad scale, large scale things, and something that I think would be super interesting in incorporating, and this is just more going into that, and like if you want like, because it took me like a week to understand this, so if you have questions, please feel free to ask me. We're kind of looking at like how it looks like with alpha diversity, so very diverse within a community, and then beta diversity, so very diverse between communities, because that can have different implications for invasion too. But what I think would be super interesting to also incorporate is vouchered specimens and observation records, so we can also have information from past field surveys and see how we can incorporate that to see a longer scale of invasion in these areas. So yeah, and after like all of that is when we want to go up to the satellite level and see how we can look at fuel loads, water efficiency, and productivity and stability of an environment after we have these other two layers that are kind of going up into it and having more data within that. It's kind of like subpixel analysis, not really. It's a very new thing, but yeah, that's just kind of the short of it. I know that was really fast, and please feel free to ask questions, but I would like to thank Laura D, Elisa Van Cleefit, and Alana Ravello. They've been helping me the most with this project, and also I want to thank my best friend Annika Jonker, who is from South Africa and has actually been answering a lot of questions that I have about the area and helping me translate some research that I find. So yeah, that's kind of the gist of it, having imagery at multiple scales, seeing what we can garner from each level to understand how invasion is impacting the environment. And yeah, I'd really like to incorporate more natural history records for a fuller, broader view of these areas. So thank you.
I have one quick question.
Yes.
So are there maps, are there images of, they're actually Landsat type images that go back, okay, so I'm looking at a specimen that was collected in 1929 or 1950. I could get the right map to overlay that and look at what you're talking about, where that specimen was then, as opposed to where it was now.
So the Landsat, the first Landsat mission launched in 1967, I believe. And like now we're on Landsat 9. So you can go all the way back to 67, and you have to do a couple of algorithms because the resolution for like Landsat 8 and 9 are much better than for Landsat 1. But yeah, that's actually something that I'm interested in doing, is like harmonizing or linking records with their respective time frame of when they were collected to Landsat imagery. So
You know, Mark heard that for black and white.
Mhm.
I don't remember when the transition to color happened, but it seems to me that would be a
yeah, so
thing to know, so that you can match a collection to the actual physical landscape at the time. That was great talk. Thank you.
thank you.
There. Yeah, what is next? I'll put it, yeah, I'm gonna put. Okay, that's great. Okay, and next is Whit Schroeder from the University of Florida. And feel free to take it away.
Great. Hi, everyone. Thanks for having me. I'm going to be talking about some of the work that I've been doing, also with remote sensing data. but with a more archaeological application. I do collaborate with other researchers as well, who are working in more of a sort of earth sciences approach. But I'll be talking more about archaeology. And then to skip ahead, just.
I need to click it over now, just to get that price. Not sure.
Great. So I really liked what David started off saying today about how we're sharing these great technologies, but we always have to keep in mind what we're going to be doing with them, what questions we're asking, how it's informing our research. And my area of research is primarily in landscape archaeology. So this is looking beyond just what we call a traditional site-based approach, where we have a specific archaeological site and traditionally in the past, it would be excavated and the objects would be analyzed and that kind of detailed work, which is still very important. But looking outside and beyond just that site-based approach and looking more broadly at the landscape surrounding sites, understanding the ecological context of where people lived, looking at the ways that people traveled across the landscape, looking at the ways that people in agrarian societies, how they worked the land, in hunter-gatherer societies, for example, how people moved across the landscape and looked for resources. These are all things that are really interesting to me from a landscape archaeology perspective. So the tool set that I use for the most part is an airborne laser scanning tool. And I'll be talking about different ways that we've been using LiDAR in airborne techniques from airplanes and from drones. But essentially it's a scanning technique that allows us to map the surface of the Earth with very high accuracy and high precision. And for archaeology, we really like that high resolution LiDAR data where we can begin to see what we call archaeological features or features of the built environment that were constructed and that have been modified over time as well. We're really looking at what we call a palimpsest, where we're looking at the landscape as it is today, essentially, but we're interpreting what we see today and trying to interpolate that and understand what was going on in the past. And this is just an example here of a type of hillshade model, just one of these kinds of derivative models that we can move from the elevation material. The really critical use of LiDAR for archaeological purposes is, of course, for mapping the bare earth or the terrain. And so this would be a landscape that at one time, that today has vegetation very likely, but we're able to filter out and classify these different parts of the landscape so that for the archaeological application, we can look just at the terrain to understand archaeological features. LiDAR was used, has been used in archaeology for some time now, but one of the probably earliest and most well-known uses of LiDAR was in the Maya region, which is where I work. And this is really southeastern Mexico, parts of Central America, associated with the Maya civilization, the ancestors of the modern Maya people today. And a site called Caracol in Belize was one of the first uses of LiDAR to map an archaeological site. And you can see we get really good resolution. We can see the pyramids, which of course are very large here, but we can even see these kinds of smaller features that are agricultural terraces built into the hill slope. And we can begin to look at the smaller constructions, which would have been the houses of the farmers. And we can see their reservoirs where they excavated to create these reservoirs for holding water, were very important in this environment where we have prolonged dry season. And so this has been sort of highlighted as the really first successful use of LiDAR for archaeology in the Maya area. The work that I've been involved with, it's actually a data set that's not too different from what we were just hearing about from a NASA system called G-LIGHT, which involves a LiDAR sensor, a thermal sensor, a hyperspectral sensor, which is where you get the acronym LIGHT, LiDAR, hyperspectral thermal. And so some of the data here that we have are these kinds of linear, sort of more linear types of surveys. The orange ones here are larger sort of coterminous surveys, but the NASA G-LIGHT data was a vegetation biomass study. that essentially just did a single flight path across these parts of southeastern Mexico. And so we've been adapting that for archaeology. The area that I work in mostly, just to give you a visual of what some of these places look like on the ground, Palenque is probably pretty well known to people here. But it's really this kind of part of the western part of what we call the Maya lowland tropical environment that surrounds that site of Palenque. These are some of the data sets that I've been using. As I mentioned, the G-Light, and you can see that this is a pretty narrow flight path. There's no scale on there, but the flight paths are about 300 meters wide, but they cover thousands of kilometers in length across southeastern Mexico. And then another data set that I've used, this is a collaboration with the National Center for Airborne Laser Mapping. And they're based, I believe, at the University of Houston currently. And so that was a survey that was planned with them. And so we actually, the archaeologists, were involved in planning the actual survey area, whereas with the G-Light, we're just sort of repurposing a legacy data set. And both of those are LiDAR systems mounted on an airplane. And then the third one is another data set that I've used that I've collaborated with researchers here at UF. They call their group Gator Eye. And they have a drone-based LiDAR system. And so again, that was a system that we planned with them, a study area that we planned. But of course, being mounted on a drone, we're more limited in terms of how long the drone can stay up in the air with battery power. So we can only map much smaller areas. I believe this is probably about 3 to 4 square kilometers, whereas this is maybe 200 square kilometers. And then this in total is, as I said, 300 meters by thousands of kilometers in length. And this is one of the projects that I've been involved with. The study was originally a RED Plus study in southeastern Mexico, and that's where the data set originally came from. And so this is kind of what we're looking for. The first step is always to kind of see what's there, right? So to identify and to annotate the features that we have. And so you can see, and again, this is one of those hillshade models. So this is taking the elevation data from the bare earth, and it's doing an algorithm to make it look to a human eye more like what the landscape might look like in that kind of bare earth sort of model. And you can go through there, you can start marking up what you're seeing, these little sort of plaza spaces with structures surrounding them, agricultural terracing, usually right along the hill slopes following the contours of the hills. And then these other interesting types of features, these are probably walled features of some kind, either with stone, most likely with limestone, which is local to the area, or perhaps some of them are earthen, built out of earthen materials. But we're seeing perhaps some agricultural type of infrastructure here. And then we've expanded this with the drone-based system here, again, through UF. And in fact, some of these flights with the drone-based system were done at the exact same time as the NCALM airplane-mounted system. So we're literally setting up the drone here, and that right up there, that's the drone, I believe. And then I believe that's the airplane they're flying at the same time. So we have really good data in terms of the exact same area being mapped at the exact same time with these two different systems. So these are some of the more, some more of the areas. So this is that G-Light data shown here. And then this is just kind of what we've been adding with that NCALM data set, which is our own sort of system that we've, our own study area that we've designed. And then these areas are the drone that we've added in there. And then we've just gotten more data, and we're just getting more and more over time. So we got a lot of data right now that we're sorting through. This is an example of one of these contexts where we might take our G-Light data, which is that transect, and we'll pick a spot where we want to get the wider context of that transect, and we'll do a mapping mission with the drone to expand. We work off of the point cloud as well, so I'm showing you some kind of raster derivatives, but just to be clear, the point data is the kind of raw data which is really coming directly out of the LiDAR mapping mission. And then anything that we come out of that, that's an interpolation of it. So it's sort of like a mesh that we're creating by triangulating those points. We can look at these changes over time with these two systems. So the G-Light, this particular mission was flown in 2013, our drone was in 2019. So we can do change detection, we can track destruction to the archaeological resources. We can also do change detection with vegetation as well, and see any sort of deforestation over time. So some of the takeaways that I just want to emphasize is that we're just sort of in the middle of this process. And so we're doing most of this work remotely right now, but the goal is, of course, to do ground truthing and to do work on the ground with communities, and to be sure to integrate this work with people living in the region today. And collaboration is very important. And thinking about how these data can help the modern communities in the region. And then a critical sort of project is to move beyond just using LiDAR in archaeology for site prospection, which is just looking at the LiDAR data and finding interesting things and identifying archaeological sites, which is very important, but kind of moving beyond that. And now we have all these large data sets. How can we automate some of this? How can we use machine learning? And how can we start to understand the broader socio-environmental context of some of these data sets? I just have a couple examples of work that I've done, sort of looking at different environmental variables, social variables, and comparing them with settlement density that we're getting from the LiDAR. And these have been published. If anyone's interested, this was a sort of research project that was to look at kind of the variation in structure size and looking at the Gini index of inequality compared to these different variables, and then looking at physiographic regions as well, understanding that environmental context. So I just want to thank all of these funding organizations and of course all my collaborators for helping me do this research. I look forward to the Q&A if there are any questions.
Thank you.
It's only screen sharing.
Thank you. Okay. Should work. Okay, got it. I'll just sit right here. There we go.
Wow, can you guys hear me?
Yep.
Okay, nice. So first of all, I'm assistant professor at Digital World Institute, and I know nothing about biology. I do not want to sound stupid, so I just literally Googled up, archaeology and paleontologist. I learned, okay, these are two different things. So I apologize in advance. I might say something that's completely stupid, so I apologize. But first of all, let's see. Is it? Okay, so today I am going to talk a little bit about myself and what do I do in our department. And and actually my students develop VR games. So in collaboration with Florida Museum, we have designed and created virtual reality game. Anybody here played a VR games before? Oh, nice. Okay, so I have a nice audience today. And so I would like to talk about a bit of what I would love to do collaborating with the Florida Museum in the future. And we'll talk about the tool I use. First of all, in our department, it's called Digital World Institute. We have like 70% of students coming from College of Art and 30% coming from mainly computer science. So what do? We create video games. These are the work that my student did for the class project, you can see they are obsessed with the battle. Actually, these are the good one. The one they actually created was like really, bloody, violent. They are obsessed with creating horror games. So I suggest, instead of creating a robot battle, can you create some more educational? And this was the content which I'm actually quite proud of. And they also created like, so this is the work they did with the dental simulation. So this is actually haptic feedback. You are going to feel the vibration as you are trying to simulate the dental procedure. Another work here you see is another of my students project over the COVID. So it's actually visualizing how your COVID virus spread your body and it's interactive. But still, I kind of really wanted to, give them a chance to create some really impactful work. And over the last summer, I found JAG. I literally just like Google, photogeometry, UF, and Florida Museum pops up. And I found that Florida Museum has like tons of this amazing asset. If I'm going to create this from scratch, because it's going to be really time consuming, I was really impressed with this existing asset. So my motivation is, can you utilize these contents and put it in the context? Because in VR, you can swim with them. You can actually go to Maya, ancient time, and explore how the site will look like. So that was the idea. And I teach VR classes every fall, and using my power as a teacher, I force my students, no, now you guys should create something with the Florida Museum. So I asked them to explore the Sketchfab and the Morpher source of Florida Museum, and they came up with, and thanks to the Edward help, we came up with 4 different ideas. The one is underwater. Well, this is a Florida, and it's about the dig site, and the insect, and the last but not least, butterfly rainforest. So let me kind of go through this, each project. The underwater, we narrow it down to 10 different specimens that we want to include in our game. The question is, how are we going to include this? Our target audience was children who visit the Florida Museum. How can we make this fun activity? This was my student's idea. Anybody play Pokemon Snap? This is really fun. The basic idea is you explore the world and you are supposed to catch a Pokemon. You can even see the evolution. Pikachu become a Raichu. I know it's not really scientific evolution at all, but the idea behind it is you are going to, study the concept of evolution, and you are going to learn like each Pokemon, like the size of Pikachu, what kind of power it has. So this was the idea for the underwater. For the dig site, Jack shared this amazing dig site, 3D scan of the dig site. So our idea is you are becoming a paleontologist, I'll say. I feel like these are 2 different things, paleontology, archaeologist. So you are becoming a paleontologist, and I'm not really quite sure what this is. I want to say dinosaur, but you have to put the puzzle of the dinosaur and it becomes alive and it moves. The insect was, actually that was Edward's idea. I learned from him that bees play a really important role in ecosystem. So here in VR, you become a bee. You fly around the Florida nature and you collect the nectar, honey in the flower. And the last but not least is butterfly rainforest. Each of the content section are YouTube videos, so I hope the link worked. But anyhow, when I was asking my student to create this, one of the problem here is that this is a proprietary item. So these are massive file size. Each file was like 2 GB or sometimes 1 GB. The really important thing was we need to optimize the mesh size. But the problem here is if you optimize this, all the textures are not really aligning. So they need to remap the texture. And the last part is animating. Because these are the static objects. Actually, I also asked my student to watch tons of video. I do not know the name of Brazilian electronic ray. I just asked them to watch the video of its movement and try to simulate its movement in animation tool. That resulted in 4 different games. So here is the one. I'm not really quite sure I can play the YouTube link. Yeah, there you go. Just voice over. Okay. There we go. No, this is supposed to be like AI tool. We want to include the ChatGPT here later. So this tiny little cute friend is going to teach what shark around, what kind of sea creature you are going to see around you. And yeah, you are supposed to take a picture of the shark as you find it. If you navigate it, let me just kind of move through. So these, you are going to find this shark and this little. You get the idea. So the idea is you capture the images of them. You are going to learn about this. And let me go back to our PowerPoint slide. This was it. And this one basically is, I'm actually going to skip it given the time. You are becoming a paleontologist. You dig the fossil sound and you are going to see the fossil of the gator. And you are going to put these into pieces together. Another one was, yeah. Oh yeah, that's and other thing was like, this is about the, you become a bee, you fly, you are going to do this to collect the nectar, and you need to actually go to the hive to meet the queen bee. That was the flying simulator. And the last map of list, here I actually put up have the website. So you can actually see the entire design process in my students' work. So because this is in VR, you can actually turn different time. I believe in the current set, it's the glass ceiling, so it's so just like the daytime, but you can change the night or daytime, and you can learn about its butterfly. So the goal is you give the banana or apple, and they are going to come fly to you, and you are going to learn information about Boomorpho, where do they live, and things like that. Okay, so that's actually for a game that my student designed and created last fall. And because I'm teaching VR class again, I am now thinking about, now let's create some AR experiences. I think the limitation of the virtual reality is you need to wear the headset. There is a heightened issue. And these are the shared devices. That means you cannot just bring your phone always, but the benefit of the AR is that these are the mobile-based device, so you do not need to provide the devices at the museum. The one thing I'm trying to do, brainstorm is, using a photogrammetry technology, we can create the object-based detection. What do I mean by this is, actually, the Met has already implemented this last year. Anybody knows what I forgot the name of it, Roblox? Game Roblox? No. The basic idea is you are going to decorate your character. You go to Van Gogh's picture and you get their t-shirts and their hats, and you can decorate your character with those assets. And something like we can actually do is stuff like this. If we are going to use a photogrammetry, we can create the object-based AR, not just based on the QR code. You move your phone, you detect the object, then boom, you are going to have some sort of information. That's something I do not really know, but I would love to have a further discussion with the Florida Museum so that we can have some educational contents targeting public audience. That's pretty much of it. And yeah, I think that is pretty much of it. A question?
Yeah, so it is now the Q&A. It's time for Q&A. So if anyone has any questions, throw it into the Q&A, or you can raise your hand and we can unmute you. And it looks like JJ, we have a question here.
Oh, yes.
I have a question for how do you make VR in augmented reality more accessible for people with disabilities who aren't visual learners or are deaf or blind?
That is amazing question. That's actually one of my research topic. When it comes to VR, for people who cannot really use their hand, there are many different things. You can use eye tracking, like as based on the input, or there has been game where you use a breath, like to fire. And when the visual is something that is really hard to actually incorporate into the VR, but there has been other research that try to incorporate the haptic feedback. If you have a realistic haptic feedback, this can be actually quite tough because most of visually impaired population, they are not completely blind. They can still see actually the color. So you can actually use a VR to highlight this specific color by making color more vivid or change the saturation. So there has been actually tons of research on the topic, but I'm so happy that you asked that. Other beauty of this is I'm actually working with autistic children a lot because the beauty of the VR AR is you can provide a tailored learning experiences. So it's not just a one-way direction, but teacher, if they are coming to the museum, let's say for the school field trip, teacher can give a specific task to the individual student, and this entire experience can be tailored to what they meet their preferences or accessibility issue. But thank you.
What sorts of engines or tools are your students using for creating these experiences?
Yeah, they are using Unreal, Unity and Unreal Engine. Thanks for asking.
Have they worked with the Godot or Godot engine at all? Do you know that one?
Oh, I never heard about it, but yeah, I'd love to learn about it.
It's a free and open source alternative, and it's been growing, especially since Unity's pricing changes last year.
It's so true. Yeah, I will definitely look into that. Thank you. Any other questions?
Any questions?
Okay, then. Thank you for inviting me.
All right, let's see. Oh, we're right on schedule. Look at that. Okay, up next is Mike Webster. Okay, should be good to go.
Okay, great, thanks. So great to see you all again. Thanks for the invite, Jill et al. Slide today, so I kind of wanted to take a slightly different tact and give a very sort of 30,000 foot discussion. Actually, I'm hoping this prompts discussion about what are some of the ways that you can use digital media of specimens once you have them. So some of the talks today have been about and I actually thought this talk might fit into what Jill had in mind for the second seminar or a second workshop. But actually, after listening to a bunch of the talks this morning, I decided this actually fits in quite well. So I'm really glad I decided to talk about this. This is going to come from the perspective of the sort of collection that I run, which is quite different from a lot of the collections that you all are involved with, which involve actual physical specimens. Our collection starts with media. It's media from the get-go. So it's not always digitized media, but it is media that either needs to be digitized, and even more nowadays, it's coming in digital format from the very start. So we don't need to worry so much about the finer points about how to digitize the specimens. It's already mostly digital, and the methods for digitizing audio have been around for decades, so it's pretty straightforward. And that's why I want to talk about some of the things that might be done with an app that they're digitized. So the collection, our collection is the Macaulay Library. It's a large media library of behavior. So we have audio recordings of birds singing, we have videos of animals displaying, we have photos. And nowadays we actually have quite a few. We have over 2 million audio recordings. We have 58 million photos. And that sets up some opportunities, but also some challenges. And so what I'm hoping I can do is talk about some of the things we've been doing with this very sort of behavior, animal communication specific collection that we have, and have us discuss and think about how some of the things we're playing around with these days might be applicable to other kinds of collections that you all might be working with. Because in all cases, we start with specimens. They might be study skins, they might be media, like this tape. We digitize them, and so we develop some sort of digital media specimen or digital media representation of the specimen. And once it's in digital format like that, you can do all sorts of AI magic with it, right? So we can feed those images into algorithms that allow us to do different kinds of things. And what we're doing at the Macaulay Library is trying to think about different ways that we can use AI for curation of our collection, in particular data quality, for helping people do science with our collection, and then for engagement and education. And the kind of digitizing we're mostly doing, or the sort of machine learning we're mostly doing, is to develop models that can detect and classify birds in photos or in videos or in audio recordings. And so here we have, the algorithms have detected these two different species of bird. It's classified them, so it's identified what species they are. We can do the same thing with vocalizations. When we make a spectrogram from a vocalization, you can do the detection and classification models on those. And the way it's done is we have people who have an incredible amount of bird knowledge, like Taylor here, who really has an amazing ear, and she can go through and annotate these recordings and box every sound in every recording and say what species that is that's vocalizing. And likewise, we can do the same thing for photos. You can identify the photos, box the photos, feed those into the algorithm, and train the models, right? What you end up with. Oh, so those annotated recordings and annotated photos go to our machine learning crews of Grant Van Horn, who's at UMass Amherst, Sam Heinrich, who works with us at the Macaulay. They develop the machine learning model that gives us the output at the end for the detection and classification. And it looks like something like this. So here's a video. We're feeding this in, and it's testing the detection and classification algorithm, and doing the same thing down here with the audio. And so it's giving us detections of different species in both the video, so visually and acoustically in the audio. And so we've used the audio portion of that to develop what we call Merlin Sound ID, which is a sound identification algorithm for identifying birds from their vocalizations. Currently, it has about 1,200 species in the model from these different areas here, a lot in North America and Europe, and then a little bit in some other areas, very little in places like Australia, a lot of Asia. But we're working on that. We have about 4,000 species that have annotations that are going to go into the model now. So what are we using it for? Okay, so I want to talk again about, so we have these machine learning models. I want to talk first about curation, and the way we're thinking about using this, or the way we're starting to use it now, is specifically for data quality. Because most of the data that we have coming into our collection are coming from citizen scientists. So it's coming from amateurs who do a recording or take a photo of a bird and they think they know what it is and they feed it in. And so we have huge issues with data quality, potential issues with data quality. And it's an issue because we have, last month in February, we had 33,000 audio recordings that were uploaded. We had over 1 million photos that were uploaded. So there's no way a human being can look at all that, right? We can't have a person listen to every recording or look at every photo and say, yes, that was a red-winged blackbird, right? So that's where we're putting these detection and classification models into the upload pipeline. So when somebody uploads a photo and they have a bunch of photos of brown pelicans and they have one that's a white pelican, the algorithm detects that and says, are you sure that's a brown pelican? It sure looks like a white pelican. You might want to reconsider the species ID on that one. And so we have, we're building this pipeline that allows people to very quickly and easily change the ID on anything that they upload that gets flagged. But if they insist it's a brown pelican or whatever, then we actually flag it to have one of our experts look at it and verify. Now for research, I think there are two ways that we're playing with using machine learning to facilitate research. One is to help researchers find what they need. So, we have 60 million photos in our collection. How can anyone possibly find a photo that they need for a particular research project? So we've started using GPT sorts of approaches to use just everyday language. This is the tool that Grant's been playing with. It's not even a prototype. It's like a pre-prototype. It's almost like machine learning doodling that he's created this model that goes in. We're doing this with iNaturalist. So you can type in bird with yellow eye and it returns a whole bunch of photos of things that it thinks are birds with yellow eyes, or bird with long bill, bird with a nest. I mean, it's okay. Most birds with yellow eye, it's mostly giving things that have yellow in the face rather than actually yellow eyes. But it's a first attempt. I think it's getting there. And that really helps someone do research projects that they want to do, like figuring out the breeding seasons for birds, right? So this amazes me. For about 2/3 of the species of bird on Earth, we don't even know when they breed. And so by saying, okay, I want to go through all of the photos and look for eggs, you can do that and you come back and there are eggs for a lot of species where we didn't know when they breed. And the other thing we're doing is actually working to develop tools that people can use for research and for conservation with the classification, the machine learning models that we developed. So one example, to conserve birds, you need to know where they are, right? To conserve snakes, you need to know where they are. And so at the Lab of Ornithology, we've developed from eBird data these really detailed, precise distribution and abundance maps of different species. For every week of the year, you can know where yellow warblers are and how abundant they are in the different pixels on this map, contrasted with a map you might see in a standard field guide, right? So this is great. These are an amazing data product coming from the eBird crew. And the reason it's amazing is because there's a lot of data pouring into eBird every day. So this is one day, 314,000 checklists were uploaded to eBird. Every white dot on here is a checklist. So there's a high data density, but it's not high everywhere, right? So most of Africa, not much data coming in. A lot of Asia, the Amazon. A lot of these places, there's not data coming in. So this is where our media-based approach is what we'd like to further develop, because you can put automated recording units out in those areas where birders don't like to go. And you can have these automated recording units just record constantly. And you get reams and reams and reams of data, but then you're in the same trap where nobody can listen to that, right? So you need to use the detection and classification models. And likewise, a lot of birds are out at times of the day when birders aren't. So migratory songbirds mostly migrate at night. And so birders aren't out there at night watching them, but migratory songbirds give distinct species-specific calls while they're migrating. So you can record them flying over, figure out migration routes, when they're migrating, what kind of weather conditions facilitate that sort of thing. Put these recording units out, get a lot of data, then use the detection algorithms to actually turn those raw recordings into presence and abundance. Okay, and the last thing I want to talk about is what we've been doing to use these algorithms for audience engagement, to really get people engaged with birds, engaged with sending data to us, because it's a very important approach for us.
is something we call the data cycle. The more excited we get people about birds and birding, the more data comes into us that we can use for these sorts of approaches. And so we've really been trying to develop approaches to getting people excited about birds and conserving birds and giving us data. And so we put the Merlin Sound ID model that we've developed into our app. It's Merlin, which is an app people use to identify birds out in nature. It didn't used to have Sound ID in it. It had other types of ID tools, but not Sound ID. And now you can take the phone and point it out at a bunch of singing birds and identify what's singing right outside the museum here. I did this morning. And when we added that capability to Merlin, this graph right here shows Merlin use per month. We're getting pretty close to about 1 million users per month in Merlin. And we added Sound ID. And so now, in the last summer, we had 3.5 million people using it per month. I'm sure we'll hit over 4 million this summer. So even, in the dead of winter, 2 million people are using it. Amazing. So now if you'll indulge me, it's late today, so I just wanted to, just for fun, I wanted to play this video of TikTok. It's a compilation of TikTok videos that Allie Smith, who works at the Macaulay with me, she went through. She said, "I wonder if people are talking about Merlin on TikTok," and she found about 5 hours of the material. And so she made a couple-minute compilation here that I just wanted to play.
Cool.
Okay. So now, put that in just for fun, but also just to make a point, which is that almost nobody you saw in that compilation videos was your stereotype murder, right? That we honestly feel like we're reaching different audiences and spreading the engagement that we do by having stumbled across this idea. And what I really would think would be a fun thing for us all to talk about is how we can do this with other digital media. They're coming from other collections, VR, proofs, whatever they are. And think creatively about using those for that kind of outreach because it's gold. And with that, thanks.
Thank you.
Can you reload the page? Sure. Yeah. So if I were smart about this, I would just say, that's the day. It's an impossible task to follow that. And what I am going to attempt to do in the final few minutes that we have is to get us thinking a little bit about how we might begin to leverage really magical things that we've seen today for a bright future in collections and biodiversity. And the take home really is that if we have clarity of purpose, we're going to be much more successful than if we are just having good ideas. That is, good ideas, as this group will recognize, are fairly easy to come by. Good problems are really what is important. So I did something novel. I just wrote half of this, and then I attempted to combine some things from the presentations earlier to illustrate points. So my assertion is that if we have a clear-eyed view of what we're doing, why we're doing it, and how we're doing it, we might have an easier time of moving forward from where we are. So let's start with what we're doing. And we are subsampling a very wide world. We all recognize that as perhaps collections professionals. And this is something that is a decision that's had to have been made for 400 years now as we built our collections. What are we going to sample in this wide world? And I would normally show this in our intro course for biodiversity specimen digitization. It's my attempt to understand what we are doing as we are creating these collections and creating digital data about them. And if you will permit me, I'll just walk through this to make a few points. So we have this wide world out there in which we are going out to sample. And we're sampling data and we're sampling material samples. And we are doing that to a certain extent. So agents, us, implement protocols to produce potentially material samples, not always, and data about a subject. And sometimes the agent is human. Sometimes the humans are helped by computers, as we saw today. And sometimes the agents are just computers. We have this legacy set of data that are pen and paper. That is, we are in the process of converting that to digital data. And sometimes we skip that today. That is, sometimes our data is born digital in the field. Sometimes we don't take a material sample and instead are just working with observations. Ultimately, then, we are implementing protocols to produce information from data and knowledge from information for some important things. And Mike just went through some of those, including outreach. Protocols are an important part of this. They make possible parallel engagement of agents so that we don't have this bottleneck introduced by just one person in the world capable of collecting a particular type of thing. And the widespread value of digitization has led to a lot of hardware options and software options. Hardware in the case of digital cameras, because these aren't being created for us, to be honest. We're instead benefiting from the fact that there are a lot of consumer grade hardware out there for people to sell sneakers or record vacations or do other things. And with many of these human and computer agents involved, we've produced a lot of protocols. This is not always true. That is, we have the work that's been done assembling 3D from glass slide collections, that there isn't a bunch of protocols in that case, but there are in others. But this is really the chassis on which is built biodiversity research today. Decisions regarding agents and protocols are guided by the most compelling uses, and then we reverse engineer success. And so I think what we have today is a lot of cases, and I'm talking about the presentations today, a lot of cases where this has not been fully described. That is, there are a lot of potential opportunities with some of the cool things that are happening, and we haven't really inventoried the complete set of things good things that can come out of them. I put this in here because I just want to recognize that while we, agreed there has been digital sound recording technology for a while, it really is recent. Everything that we're talking about is recent. Even, the most basics of photography and audio recording, humanity has not had an opportunity for the use of this until very, very recently. And even more recently have we seen this inflection point. We talk about analog versus digital, and this was a study that came out in Science in 2011, recognizing that we had an inflection point around 2007 between things being analog and now things being more digital than analog. So this is a bit of context in which to think about what we're doing. I was listening for people to reflect in some kind of philosophical is the wrong word, but some deep way about what they're doing. And I heard a little bit of that. I mean, Duncan's had a reference to digital heritage. That's a really interesting concept. I would have liked to have asked him about that. And John had the active continuity. There's something there that I would love to understand, but we didn't have a lot of reflection. I apologize if I'm missing something that you said. I did step out during the presentation. The other thing that I didn't hear was nobody said digital twin. I don't know why, but nobody said digital twin. And this was something that was front and center in my mind because I had an extended conversation with an archaeologist about digital twins yesterday, or Friday, I guess. Yesterday was Sunday. But I think that there is the potential there for us to begin to frame what we're doing in a way that has some greater clarity, perhaps. Why? And the why is not necessarily, let's think beyond research, I guess is what I'm trying to do with this section. So for the workshop, we recognize that we're seeking to move this activity to even greater relevance to science, society, and Earth's biota in the future. And Jill perhaps had referenced this at the beginning. So this is a why answer. I think if we're honest, there's a certain desire to be clever, right? I am unabashedly in this camp. I attempt to do things that I think of as novel combinations of things. And this is the good idea that I was referencing before. That is, this is a smart group and we have good ideas. We think, you know what, that's cool. It could be applied here as well. It's not that you are going out perhaps and asking, so what does the world need? And then working backwards from there. I think that in my case, what I tend to do is, that'd be cool. I think that'd be cool. And then I go out and look for a problem to connect it to justify the fact that I'd like to spend some time on this. But we want to get beyond that. I don't want to suggest that it's bad to think about how to be clever. This is another thing that we talk about in the courses, and there's a bit of a valuable feedback here. That is, if you are producing research products that are valued, your reputation is increased and your ability to gain funding to continue doing that is possible. Now, there is this piece here that I'd like to just recognize. That is, we've seen impact in several of the presentations, and this is an important extension of what we do. We talk about it when we talk about broader impacts for NSF. We talked about it in the context of several things today, but it might be worth just keeping these things in mind as we talk tomorrow. So biodiversity is a central part of what many of us do. And obviously we are in a crisis at the moment, and this is one thing that might motivate what we're doing. And that can be thought about in the context of, for example, multilateral environmental agreements. This is the Kunming-Montreal Global Biodiversity Framework, which is a really interesting agreement. I don't know if you're familiar with this at all, but the agreement includes a recognition that there is there could be the conservation of 30% of lands and seas by 2030. That 30 by 30 should be a major motivator for us. That is, we have a lot of data that could be valuable in the context of identifying what 30% of the US should be preserved by 2030. I again apologize if I missed your why, but Dave started with the who, the people. Nicole mentioned outreach, educational portfolios. I like this one, hidden specimens. That was really a nice idea. Trailblazing the pipeline, cultural revitalization, which was a particularly compelling use case, I thought. Access and benefits sharing, diplomacy, predicting fire behavior, archaeological site discovery. And I'm sorry, I had to submit my talk before I had a chance to add Mike's stuff. So those are two questions we might pose. Finally, we might pose how. And how is more, it's not so much the protocol, but it's the social side of things. That is, we heard two references, and I'm going to struggle to remember who it was, who talked about scalability. Nelson you said at one point, we seek to be scalable solutions, something like that. Maybe Zach had the other one. The idea that that was about the protocol, but it's an important point. So here's a few of what I heard related to the how. People-centered, keeping people at the center of these activities, data requirements. I love that, Adam's recognition that the methods have to do these things. Fair availability not equaling accessibility, John, accessible methods, that's something that's different than accessible data or models. Do no harm to cultural samples, and science is done by people. I think that we all recognize that we're doing trailblazing work, and we really need to consider how we are going to train others to do this. And it could be that it's simply a matter of publishing well-written protocols. Some of these things are fairly straightforward to do. Others might require training. And this gets back to the accessibility issues that Ed mentioned. And we do, for example, in the Digitization Academy, the iDigBio funds have a digital imaging course. It's 12 hours long, focused on digital imaging for collections. And finally, I just want to recognize that it is really important for us to think about the ethics of scientific documentation. I don't think I heard anything come up today. There is a long history of need here. For example, we've been doing scientific illustrations by hand forever, and boy, isn't it a little tempting to, maybe complete that leaf where something had eaten into it, guessing at the margins and that sort of thing. And so there's a lot of history there, but we do want to keep this in mind. We do want to recognize that while it might be convenient for there to be a seventh leg, it shouldn't be there. You know what I mean? And I'll stop there. Thanks so much for your attention.
Stop sharing. And that was our final speaker for today. If anybody has any questions for Austin or for Mike, I think there was a Q&A from the last session too. I guess this is our last opportunity for Q&A. So we might have lost some speakers along the way, but for the most part, we've got most people here, so.
Like, it might be a somewhat selfish question, but you showed the map of Merlin's sort of expansion through the world. Last time I visited Australia, I only worked on introduced species. How far away are we from having like wildlife?
There's 12,000 or 11 some thousand species of bird in the world. Right now for photo ID, works for about 7,000 species. For sound ID, it works for slightly over. So in that sense, we're a long way away. But from it working, Australia is a particular embarrassment for me because I work in Australia and have for many years. And I just was down there in November at the Australasian Ornithological Congress, calling for increased reporting of birds. So the problem is you need about 100, 150 reportings per species to train these models. And for only 27% of the species in Australia do we have that. So the raw material coming in is very rich in some regions, surprisingly rich in India, for example, but not in other regions. And Australia is a great example of a country where you would expect there to be a lot more. So,
would it be helpful to, even if we're not using it to actually ID birds, if we can to just like collect the sound information and send it through the Merlin app?
Yeah, so I didn't talk about this, but we do sound recording workshops, planning stuff for Australia in November of this year, working with a lot of the sort of BirdLife Australia. So I just have some staff at Ecuador right now. So yeah, getting the raw material is critical. And then having the expertise to annotate, you need local folks who know the local birds to do
that. Do you know, has Merlin type information been expanded to other taxa groups, for instance, called, et cetera, et cetera? Or the feasibility even?
We're not doing it, but there are some people who
doing that, yeah. There is a lot of interest in doing it for frogs, in particular, because you don't need nearly as many recordings to train models for frogs that don't have the same kind of repertoire issues. They do have temperature dependent issues, so there are issues, but it's simpler. There's less geographic variation, less repertoire. So there are efforts that are great. But we have material. If people want to come to us and say, hey, we need frog recordings, we have frog recordings.
Quick question for Io. So I know you said you kind of like force or joke and force your students to create these projects. I was just curious, like, what's the reaction from your students? Do they love working with kind of like the natural history stuff, or would they rather be trying to do some bloody combative style?
That's the thing. I mean, as you can see here, all these ocean creatures do not live in a specific environment. Those are not scientifically accurate, but they want to create more bloody wildlife content. They love the idea of creating a shooting game. I don't know why. But it really depends. It depends on the students, because my students want to work in the game industry, but some students want to work in the research area. So if I can persuade these groups of students, then you create more scientifically accurate. So it's all about motivation. That's a good question, and still like very challenging. If you guys have any, public outreach contents or ideas, I'm again teaching the PR course this fall. So let me know. I'll love to use this free later to make some video.
Yeah, we also had a question online, but we've like already stopped for Ohio. And Elizabeth asks, what is the best way to work with VR or AR designers as museums or biologists? What time should we discuss the project, and how close should the collaboration be?
That really depends on what kind of final. For this kind of work, these are just like 5 or 6 VR experiences. So that's like 3 months work. Not just 4 work-based students or 5 students. But if what Metropolitan Museum, that's obviously a really expensive work that requires tons of programmer and designer. So that again really depends on the scope.
And just to repeat, if people online didn't hear, it really depends on the scope of the work, and like how much work that you need to do for that question.
I was wondering if, sorry. I was wondering if there's some hay to be made out of kind of combining the approaches of an augmented reality based interactive with ID, apps. You're out in the field and, I know you've got a lot of lovely pictures and things, but having an AR version of the birds that are cool and you can look around and see, get a sense of scale as well. Because one of the things that I think the digital imaging loses with a lot of that is the idea of size, right? We don't usually have scale bars in all of our images, and it's just a 2-dimensional image of a 3-dimensional thing on a screen. But with augmented reality, you actually do get a physical sense. I saw a frog map in Australia. I couldn't believe how big that thing is. I assumed it was going to be like a sparrow-sized thing. It's enormous. So, that sort of thing I think would help, could potentially help with some of these ideas. You have all of that baked in with photogrammetry and light scan methods. So just a thought.
So I disagree with it. I think that makes a lot of sense. I really feel like we just scratched the surface. What we're doing, we just made a thing that IDs birds on. That's it, right? And it's really taken off. I think it shows that there is an appetite for that, and creatively thinking ways for people to use other tools, maybe combined with Sound ID, maybe totally different, that just draws them into nature one way or another is the ultimate goal.
There is actually app I use. I forgot the name of it. So you take a picture of your plant and it detects the name of the plant and tells you, oh, you should not water them twice a week. So I kill so many plants, so that's why yeah, yes.
I did. It's for you, Austin, and maybe for the room on a more not philosophical tilt, but everyone who's worked in collections or archives, I think, has encountered ways that the collections or the material gets used that would have been completely unimaginable to the people who did the collecting or took the measurements or did the work to begin with. And when you talk about why we're doing the work that we're doing, I wonder if there are ways to harness the unknowability of what we're doing to motivate us. I mean, to motivate the work. That's not usually how this goes. Like, it's maybe the opposite of what you're saying. Like, you want to identify very clear things like biodiversity loss or policy making or whatever to motivate your actions. But there's so much that we don't know what the activity will be used for ultimately. I mean, even with Merlin, it's like you see people using it in all kinds of ways that you never would have expected, I think. I don't know, is that something that can turn into motivation perversely?
Yeah, I think so. I mean, it's a great point, and I would suggest that what might be appropriate is framing the motivation like one step up. So there might be a lot of things that are happening that you wouldn't expect by artists and fifth graders, and you have a long list. What that all represents though is what could be described as mainstreaming biodiversity. Like that could be your motivation. Your motivation is getting biodiversity in front of people and getting them to value that biodiversity. And that could be, you could be checking that box in a bunch of ways. It doesn't have to be identifying a goal that is, I'd like to see it used more in middle school. That seems like a fine kind of thing, but it also doesn't motivate a group bigger than your team, perhaps. And that's really what I think we need. I think we need, as a community, to say, if we got these 3 things done in the next 5 years, like we really advanced the ball when it came to mainstreaming biodiversity and providing actionable data on what to preserve in 30 by 30 and some third thing, that could be motivating a whole community of people who are developing methods for new novel use of digital media and using it. Maybe they're not trailblazing, but they are using those protocols and seeking to do so because they're part of this bigger thing.
I was just going to follow up on that. I mean, I always think of that as kind of future-proofing, right? You're future-proofing the collections. You don't want what you're doing in 2024 to be, useless to most people by 2030, right? So, and I think the other thing to remember is that we're copying things off of real objects. So, I think we also need to, we recognize that the cost here is the effort that we're putting in and the money we're spending on storage and the expertise the time we're taking away from other projects to create these digital facsimiles. But hopefully the specimens themselves remain and persist. And so we can prioritize rescanning things or doubling down on things. One thing that I think about a lot with long-term storage of CT data, and I'm sure that the photogrammetry folks think about this a lot too, is long-term storage of the raw data, right? The radiographs or the original photographs, which are enormously, and you probably don't need to, reuse. And so, but of course, future technology might make it much more impactful, to go back and re-extract all the data out again. You can get much more information out, especially with, machine learning techniques that allow you to keep using neural network machine learning digitally, because I don't understand either of my thing. But the idea is that, you can go back to the thing, but of course, if it costs, $100 to store that thing for for 10 years and it costs $10 to scan it in 10 years' time, you know, it might be worth, and you have the specimen, it might be worth just jettisoning the data, you know, and doing it again. So, I mean, you know, just something that I guess we try to plan around, you know, to future proof. And recognizing this isn't the forever, we're not creating the forever solution to this, right?
I don't know if maybe kind of piggybacking or leapfrogging off Ed's idea, but you mentioned something about future proofing, and then just rescanning if it's viable, so at least for CT scanning, viable if you still have the specimen to rescan. Something that maybe a lot of people don't think about is the lifetime of a specimen. For wet collections, it varies. I know that Jamie talked about earlier about formalin specimens. I've scanned some things that have been preserved in formalin that have demineralized, so the bones that they're after, they were never going to be seen in 10 years' time after they were collected because the formalin basically ate away the mineralization of the bone. So you have this researcher that's super interested in this developmental juvenile stages that they thought would be maintained over time, that you actually don't have that. So something also to consider is these temporal timelines. For CT, it varies. I'm not familiar with what those would be for photogrammetry and other things, but you know, the condition, even the conditions of the rattles and the whistles I talked to you about, they've had them since the 1800s. And there's already some damage just over time. The wood becomes brittle and it cracks. Or for me, the paint, it'll kind of get dirty and darkened, even though they are stored at the optimal best settings. You know, there's just stuff that happens over time that you can't track the progress of. So when we're creating these digital replications or these, I guess, archival records of them, or even through sound, you know, that's just something we have to consider, plus the technology changes. So something that in early CT, looking at early data sets, there's in 10 years' time, the technology changes, but the adaptations to how you like look at how to read that first recording or look at that data doesn't exist anymore. You know, you can just look at UPS drives and UPS-C drives, like the connections and the ports keep changing, but it's just something to consider as well in imaging.
And if I could just piggyback on that, I do think that this changing technology is really critically important, and it's both a blessing and a curse. The blessing part is that technologies to analyze these data will be constantly getting better. That's what one of the things I was sort of maybe inarticulately trying to say in my presentation is, you know, we're just playing around with this now. In 10 years, 20 years, there's going to be amazing tools, and having digital representations, digital images of the specimens creates that opportunity. Right? So that's the good part. And just to give an example, you know, the Macaulay Library started in 1929. And at that time, they were testing what was considered cutting edge technology, which is talkie films, right? Sound synchronized with film. So they were saying, can we actually film a bird singing? That's really the cat's meow. We can do that. That's amazing. And now look where we're at, right? It's just come so far since then, but that's where it started. I mean, that was decades before magnetic tape was invented, or before the spectrogram analysis was. So all these new tools come along and allow new things to be done. On the not so good side of things is the digital media formats themselves will constantly change. So and a lot of you already know this, but in the digital media world, talk a lot about data migration. You have to constantly migrate the data to new formats, or else, you know, you end up with a file you can't open. So that creates work. So that's a real serious consideration for us as well.
Okay, I am going to stop the webinar. Thank you so much to all of our virtual attendees and all of our speakers. And hopefully we'll be able to share some more with you in the near future. But thank you to everyone who joined today.